Okay, after the session yesterday, or two sessions on glycobinding protein and the glycans itself, so today we're going into how we analyze the glycan. So we're going from glycomics to glycoproteomics, etc. And in particular, and it's going to tell you a lot about the historical development. Okay, so this session we have three speakers, and Anne, Daniel, and Manfred, which are introduced each when it comes. So first, let me introduce Anne. Professor Anne Dale, coming from Australia, and she got her PhD in Cambridge, and then after that moved to Imperial College, and has since then stayed in Imperial College until now. And she won many awards, and is quite famous in the field. Everybody knows her, and she's going to tell you the fascinating story when we ourselves, when we are young, when we started with the mass spec, and how we use mass spec to analyze glycan. So among the awards that she got, is plenty. I just want to mention that she did get the IGO award, which is the IGO award, in 2005, and then the ICO, which is the other one, the International Carbohydrate Organization, Whistler Award in 2004, and then also she is now a fellow of Royal Society, British, and then she's involved in a lot of BBRC council. So I would not say too much, and I'll leave Anne to tell you about her work as well as all the historical development. Okay, thanks.
Thank you very much, Ku. It's a great pleasure to be here. And before I start, I do want to thank Ku and the other organizers for what has been already I arrived last night, so I didn't see it happening, but I've heard it was a really successful yesterday. And I have to say, I want to thank all of the people who've been helping speakers come from abroad, meeting at the airport, arranging taxis. It's been absolutely superbly organized, so thank you very much, Ku. Right, so we do have the first intro slide up. So for the first few minutes, I just want to say a little bit about what we are hoping to cover this morning. So I'm going to give a little bit of history to put into context a lot of the techniques we now use, but then I also, in my talk, bring us right up to date with some very recent work we've been doing to give you an idea of where we are from a pure glycomics perspective. My talk will then be complemented by the other two talks that will branch into glycoproteomics and also imaging. So the second bullet here gives an indication, it's in your booklets as well, of the topics that we're going to be covering. So I've forgotten actually what I meant to do. So this will change the slides as well, hopefully. So just to highlight what the talk titles are, as I said, I will talk a little bit about the history and the current status. Daniel is then going to give the Hitchhiker's Guide to Glycoproteomics. So hands up those in the audience who know what the Hitchhiker's Guide is. Aha, there's a few. Wow. Okay, so I'll leave it to Daniel to actually fill it all in. And then Manfred will be going to the higher level, as it were, of intact proteins, high throughput, and spatial glycomics. So with that, unless there's actually, I was going to also, just before I get started, try to get a feel for the audience. So who in the audience has never used mass spectrometry? A few, excellent. Who in the audience uses it every day? Ah, wow. A few as well, right, so everybody else is in between. That's great. So I think that probably sets the scene for the morning. And our purpose, Daniel, Manfred and I, are to make sure that all of you feel that you know everything by the time you leave at the end of the session. Now, of course, it's a myth that anyone knows anything, including us. But the idea is that what we're going to try to do is to tell you how and why. Obviously not with the great detail that's needed to do it, but at least if you know how and why, that you then know what to read or who to talk to, okay? So that's the plan. We'll have roughly, assuming we all keep nicely to time, roughly 10 minutes each of us for questions and discussion, and Ku is going to keep us all in charge in terms of timing. The clock, I gather, doesn't work at all, the nice big green one on the walls, so we will estimate as we progress. Okay, so if I could have the first slide for my talk, Thanks. Okay, so what I'm planning to do, as I said, and I'll have a slide in a minute covering the topics so that we don't all get off track, is to talk a little bit about the history of where we are and then bring us right up to date. This picture here, for those of you who haven't been to London and visited Imperial College, shows the South Kensington part of London where Imperial College is located, and it is essentially all of this here with lots of museums around and a lovely park at the top. So do come and visit. Anyone is always very welcome to come, as happened when I first started out in Glycomics, as I'll show you in a moment. So this slide here is just highlighting, for the few of you who put your hands up that you haven't done mass spec at all, that there are lots of ways of going from having a sample to getting data that is meaningful in terms of the glycan composition, where the glycans are on proteins, etc. So all of these pathways, and this is a very simplified set of pathways, can be very confusing, especially if you're trying to understand from the literature, because everyone has their favorite pathways and will always promote those pathways. So the idea this morning is to give you an impression of which you should choose and what is potentially relevant for the project that you're involved in. So what I will be focusing on specifically is how can we take a sample, whether it is a band on a gel or whether it's a purified glycoprotein, for example, perhaps an immunoprecipitate that's been purified, or indeed whether it's a population of cells or organs and tissues. How can we take any of these kind of samples and put them through a realistic, from an experimental point of view, set of procedures that allows a wide variety of people to use these technologies so that they aren't limited to having to go to a very specialist laboratory like ours, that you can actually try it yourselves. So this is going to be the focus of my presentation today. And the topics I will be covering, firstly, as I said, historical perspective, particularly highlighting permethylation as a technique. It's a very simple chemical technique. Many people in labs that aren't set up to do chemical experiments, in other words, biochemical labs or biology labs, are often reluctant to try permethylation, but it is a very straightforward procedure. So I would encourage you, if you are venturing into glycomics, to try it. You'll be potentially very surprised. It actually works very well. To highlight the methods that we're using in the applications, I will show some snapshots, a few slides, just covering a few of our projects over the last 10 or 15 years, just to highlight a couple of key points about the methods. And then to bring us up to speed with what's actually happening, as it were, today, the last section of the talk will focus on one of the projects that we're involved in at Imperial, looking at the potential importance of glycans in premature birth. And as I pointed out at the bottom here, although my talk will nearly all, apart from the very beginning, focus on mammalian glycoproteins, the methods are applicable to any glycoconjugate, provided the sample can be taken out from the top right-hand corner of the previous slide and put through a permethylation process. Okay, so a little bit of history, a bit of history of my life, actually, that goes back to the 1970s. And back in the 1970s, mass spectrometers could be used for molecules that were volatile. Pretty much everything that was being done in mass spectrometry at that time required the sample to be turned into a volatilized sample before it was ionized. And that excluded, essentially, pretty much all biological molecules other than the small metabolites, steroids, etc. And in the late 1960s, early 1970s, Howard Morris, who initially had started at Leeds in the UK, actually as a PhD student, with ideas that a mass spectrometer could be used to sequence peptides, peptides, which was an unprecedented vision at that time. By the mid-1970s, he had pioneered the development of mass spectrometry equipment which was capable of analyzing high-mass ions. And at the same time, he helped to pioneer, and in particular exploited, the technology of Mickey Barber and colleagues at the University of Manchester Institute of Science and Technology, a technique called fast atom bombardment. And I'm going to focus the first 5 minutes or so of my talk on fast atom bombardment, because even though it's hardly used now, everything that we now do in MOLDI in particular based glycomics, but also some of the electrospray glycomics, has its roots in fast atom bombardment, and all of the methodology has its roots in fast atom bombardment. So the really key thing was that by the time the early 70s came along, and particularly from about 1975 onwards, it became possible to look at large polar biomolecules like glycans. And all of modern glycomics is based on this molecule. So this is an amazing molecule. It's called, or at least I think it's an amazing molecule. It is called methyl glucose polysaccharide, or MGP. And Professor Clinton Ballou, who passed away now sadly, but was one of the absolute giants of carbohydrate chemistry of the last century, he was working on a whole variety of polysaccharides, glycans, glycolipids, etc., from Mycobacterium sphegmatis. And the reason I think this molecule is amazing is because it is a unique oligomer in that it is not a mixture. It is simply a defined number of sugars. It is modified at the reducing end with glyceric acid. And before it became MGP, i.e. when it was in the Mycobacterium, it actually was a glycolipid. And Clint had removed, or his lab had removed, the acyl group that was on the glyceric acid, and also acyl groups that were on these sugars here, the glucoses. And I'm highlighting that because I'll come to that in a moment. So Clint came to my lab having learnt through the grapevine that we had this technology that would analyze high molecular weight, or fairly high molecular weight oligomers. And here is indicated what he believed was the composition of this molecule. So at that point, so the molecule was predicted, that composition was predicted to have a molecular weight of just about 3,000. And the technology at that time for high sensitivity, the limit was M over Z 3,000. But pretty much nothing had been analyzed above M over Z 1,500. So I wasn't super optimistic that it would work. However, it did. And it turned out that not only did it worked with absolutely amazing sensitivity and abundance of ions that had never been seen before in any mass spectrum of any kind of oligomer at that time. And this is a hand drawing traced off photographic paper, because that's how the data was acquired at that time, traced off by me and numbered the molecular weights of all of the fragment ions. The molecular ions are down here. Absolutely stunning data for that time. Nothing had been seen like that before. All of the isotopes clearly being shown here. Molecular ions plus sodium and plus potassium. And these masses corresponded to the original structure, which was everything minus that residue and an additional residue on here we showed. So in other words, we were seeing 2 additional sugars. And we were also seeing masses of fragment ions. So I was absolutely delighted. In fact, over the moon we were. We had just never seen anything like this before. So it was very, very exciting because we knew the exact size and we knew that we would be able to apply this probably to a whole variety of molecules. But my enthusiasm was dampened by Clint telling me that what I said was the structure was wrong. And essentially, what we realized by seeing this original data on a molecule which was naturally tagged, it was tagged on these inner glucose residues by these methyl groups at position 6, also tagged on the capping sugar here. When I assigned the spectrum was telling me that the non-reducing end was a methyl hexose, followed by another one, all the way up to 11, and then the first non-methylated hexose. And of course, that is not the sequence here. The sequence had been shown unequivocally by NMR in Clint's group. So what we knew immediately was that the molecule was falling apart at this point here, and therefore it was this part of the molecule that was being seen with this as the apparent non-reducing residue. So we learned a lot from that simple, well, it wasn't such a simple analysis, but the spectrum that was so beautiful and very easy to interpret. We learnt, firstly, that increasing the hydrophobicity made these sugar polymers fly, as it were, much, much better in the mass spectrometer than had ever been seen before with hexose oligomers, which you could buy from the companies and try to analyze. They just didn't work. Glucose polymers just didn't work on these techniques. We also knew immediately that the fragmentation was very strongly influenced by the presence of that hydrophobic substituent. We knew that fragment ions would arise from double or multiple cleavages of these kind of molecules, and therefore we would need to know whether they were multiple cleavages in order to interpret the data unequivocally, because if we had a branched glycan, we wouldn't know whether or not the branch had fallen off, whether it was still there, and so on. We also knew that having the reducing end tagged, which in this case it was tagged with glyceric acid, was really, really useful for telling the ends apart. I mentioned just a few minutes ago that MGP comes from MGLP. So MGLP has a whole variety of lipids on it, and it moves its molecular weight to over 4,000. So we hadn't initially thought we would ever be able to analyze MGLP. But having got that stunningly beautiful data, we decided to look at MGLP, and we decided to try it in the negative ion mode because of the glyceric acid. And we obtained extraordinarily beautiful data. I haven't put a picture up. You can look at it in this paper if you want to see it. And what we realized by looking at it in the negative ion mode was that if we had the negative charge located at the reducing end, as it was in that glyceric acid, then that would direct the fragmentation and include an incredible variety of cross-ring fragments, which would allow us to learn something about branching and linkage. So the extra lessons we learned from looking at MGLP and MGP in the negative ion mode was that if we have a negative charge in the molecule, perhaps more than one, that can be extraordinarily helpful in giving us well-defined cross-ring fragments and fragments that are complementary to what we would see in the positive ion mode. Overall, we knew that hydrophobicity was really important. We knew that the cross-ring fragmentation was more prominent in the negative ion mode, which is why so many of the mucin people, quite rightly, use the negative ion mode if they're not fully derivatizing their molecules, even today. Having the reducing end tagged with a negative charge ensured that we had dominant reducing end fragments. It also told us that if we were to put a tag in that would have a positive charge, then that would do the same. And that, again, is the foundation of a lot of the tagged molecules that are currently used in high-sensitivity glycomics. So the next steps, we kind of realized, would be to make the sensitivity even better by increasing the amount of hydrophobicity. And that's where the full permethylation came into play. And I was very fortunate that soon after Clint was in our lab, we were visited by a whole variety of people with samples that gave us the opportunity to look at pretty much every class of glycan and what we could analyze with the methods that we had available. Plant cell wall, polysaccharides, for example, bacterial glycoconjugates. But what I've chosen to show you today, and I found particularly exciting at the time, was the introduction to mammalian glycoproteins. And this was done through wonderful colleagues, Minoru Fukuda and Michiko Fukuda, from what was then the La Jolla Cancer Research Foundation, when they provided me with samples of these absolutely wonderful molecules, like these long polylacnate chains on what is called band 3, which is the blood group carrying glycoprotein of red blood cells. And we realized that the fetal and the adult were different in that these were linear and these were branched. The little I, big I antigen that had been characterized by others, including Sen Hakamori and Tenfezi, with respect to antibody binding. We also looked at these lovely molecules with lots and lots of fucases on, and in particular identified Salar Lewis X here on these cancer cells, but also on neutrophils, which of course predated the discovery of the selectins, so it was wonderful 4 or 5 years later when the selectins were characterized. I just want to point out the key down the corner here for those who are not familiar with the colors and the symbols, because I'll be using these throughout, with the exception of a couple of early slides where we had different colors. Okay? So, what did we learn from looking at all of those beautiful molecules and plant cell wars, etc.? We learned that the sensitivity was increased by orders of magnitude by permethylating the whole of the molecules. We were also able, especially if it's a eukaryotic glycan, to define unambiguously from the molecular weight the composition, because the individual permethylated sugars have very distinctive masses, more distinctive than the non-methylated. So there's no ambiguity normally in these compositions. We knew that there were very predictable and totally reliable fragmentation patterns. And what that means is that we can use not only the fragment ions, but also the absence of fragment ions to tell us about structure. So if we do not see a fragment ion that corresponds to a particular composition that we're looking for, we know that it isn't there, or at least it's not there at the sensitivity. And we always have a pretty good idea of what sensitivity we're working with. There's absolutely no ambiguities for multiple cleavages because of the internally derived fragment ions of fewer methyl caps. So if you're a newcomer to the field, I would urge you to read this review I wrote in 1987. I know it might seem very old, but it actually has all of the basic principles of FabMS, which are all applicable to moldy MS today. I think it is behind a paywall now, but it's not that expensive to buy it. There's a few coffees. So with respect then to the history and jumping up almost to the present, the big change in the 1990s was the slow phasing out of fast atom bombardment with the introduction of MOLDI and electrospray. But it took another 10 years for MOLDI to replace Fab, largely because the early MOLDI instruments did not have any accuracy for the fragment ion assignment. So if you're reading older papers and you're puzzled by the masses not being right, the fragment ion masses were not accurately assigned in the early MOLDI instruments. And in fact, it was also quite difficult in MOLDI in the 1990s to get MSMS fragmentation anyway. So there's a lot of words on here. You've perhaps had time to read it. But the bottom line is everything that we had done with Fab is applicable and still is applicable to the other ionization techniques. There is another review that we wrote in 2001 which summarizes essentially that transition and particularly introduces a lot of the glycoproteomics that was being done at that time. So for newcomers, these are my slides for introducing MOLDI and actually how it differs from Fab. In Fab, if you remember that gorgeous spectrum, the fragment ions and the molecular ions are produced at the same time. You can do MS2 experiments, but actually most of the time you didn't have to. The difference for MOLDI is that there isn't enough energy imparted on ionization usually to give the fragment ions. So the fragmentation is uncoupled from the molecular ion formation. So when you see terms like mass fingerprinting or mass mapping, that's the first stage, where the sample is ionized, gets a little bit excited, but not too excited, and you see the molecular ions at superb sensitivity. In order to see the fragment ions, you have to give it energy, and this is usually called collisional activation, and essentially you get beautiful fragment ions that tell you about sequence. And as I mentioned before, what the molecular ion tells you is the composition. So for example, if we have a molecular ion at 2,967 when it's methylated, this is an unequivocal composition as long as it's not a bacterial glycan. Okay, so 2 sialic acids, 1 fucose, 5 hexoses, and 4 hexanacs. Now, if you're completely new to glycomics and glycobiology, you probably won't know why that can be translated into these 2 structures, but don't worry, at some point you will learn that. So basically, this is N-linked. This would be an N-linked glycan we're analyzing here. An N-linked biosynthesis is very well defined, and in fact, the early roots of defining the biosynthesis of N-linked glycans goes right back to the 1960s and 70s. And the bottom line here is that the sialic acids are on the ends of the antenna, so we can put one on each, usually. The fucose, however, can go on the core or on the antenna. And the MSMS experiment unequivocally defines whether it is a mixture or whether it is solely on one or the other from the fragmentation which takes place as a single cleavage predominantly on the reducing side of nitrogen-containing sugars, so on the sialic acid, which has a nitrogen in it, and on the blue squares, which are hexanacs. And we get very, very clear fragment ions from showing the charge on the reducing end of the molecule up here or the non-reducing fragments down here. And that is the basis of moldy glycomics. Mass fingerprinting of the molecular ions and these beautifully clear fragment ions. So even when life gets complicated, we can still come up with answers. So this is a typical moldy spectrum or series of panels showing the masses. I know it's small, you're not intended to read it. We're showing the masses all the way from about 1,000 down at the bottom, all the way up here to over 5,000. In fact, it keeps going to over 6,000. And what I'm showing here is that you can see that there are hundreds of different glycans. In fact, there are thousands of different glycans. And these are all incidentally on a single protein. It's on a protein called glycodelin A, which is isolated for amniotic fluid. And glycodelin is a relatively small protein. It's only about 18 kilodaltons. And when we first analyzed this by both glycomics and glycoproteomics, we looked in detail at the glycosylation at each of the occupied sites, which are 63 and 28. This site is not occupied, even though it's a consensus sequence. So there's Fab data from the 1990s, together with electrospray data, that was done, and then later MOLDI was used, which is more sensitive for the higher masses. And this paper here, if you're interested in looking at this, shows that glycodelin has a very rich variety of epitopes on its antenna. It can be bisected or not, core fucosylated or not, and it can have 2, 3, or 4 antennae, or it can be hybrid. So all of those combinations are found in that single glycoprotein. Another key development of glycomics occurred in the mid to late 1990s, which was the development that allowed us to do glycomics even when we had a really crude sample, i.e., we simply took chunks of tissues to see what the glycan profiles were looking like. This work was done in collaboration with Jamie Marth at UCSD at that time, and Mark Sutton-Smith was a PhD student with me at the time who was working on this. And I just want to show you a tiny bit of data from the knockout mouse of alpha-manosidase 2, which is the enzyme that cleaves these 2 mannoses, the 3,6 mannoses, off the precursor for complex glycans. And here by the way, is what the kidney in that mouse looks like when the mouse dies, and it dies prematurely, although not that early, actually. It's premature, as it were, in terms of its death, but it does survive quite some time with a pretty non-functioning kidney. And this is data where we have different colors, if you've noticed the colors, because this was before the colors were decided by Essentials of Glycobiology and the Consortium for Functional Glycomics. So there was a swap of the red and the yellow. So basically, here's the wild-type kidney of the mouse, and here's the knockout kidney. And as you can see immediately, it is obvious what the changes are. There are changes to hybrid structures, loss of these high molecular weight, not entirely, which is probably why it survives. And basically, there's all of these rather interesting novel structures that this mouse is making. If you're interested, the data is in that publication. So just to pause for breath before we come on to a couple of additional examples, and I hope I'm doing all right for time. How much longer have we got? Yeah, okay. I'm just reproducing the earlier slide with just a tiny bit more detail in case those who are more experienced are wanting to know what happens in between here and here. That essentially we reduce and carboxymethylate the sample, which can be a real complex mixture, or it could be a nice pure glycoprotein, in order to make it readily digestible with trypsin. Then, once we've done a trypsin digest, we remove the N-glycans with P and GAs F, and we remove the O-glycans separately as a second step with reductive elimination methods. These are very standard methods that everybody has used in the structural glyco world for 20, 30, maybe even 40 years now or more. Key steps, though, are the permethylation before we go into the mass spec and the data analysis. There is a lot of data that was accumulated over a period of 10 years or so under the umbrella of the Consortium for Functional Glycomics that was led by Jim Paulson. Jim is here, is he? Jim here? Yes, no? Maybe not. Saw him last night at dinner. And again, if you want to get an idea of what these glycan profiles look like, they are publicly available on the CFG website. And what we did under the CFG was to look at mouse and human tissues and cells with a focus on the immune system, not entirely, but so many of the immune cells data are in there, but also quite a lot of tissues, so that you can see what kind of glycans are being made by different organs. And I just want to show you a tiny bit from neutrophils, just again to give you an idea of the data. So neutrophils, as I mentioned earlier, had been characterized way back in the early 1980s by fast atom bombardment. What MOLDI allowed us to do was to look at all the minor components with much, much higher sensitivity. So the glycan profile, again, this is a standard glycan profile now, if you like, when you look at it. In other words, lots and lots and lots of different glycans. And if you want to look at it in detail, that's the publication. But what I want to point out is that you might be looking at all those cartoons and thinking, oh, do I have to spend all my life actually drawing them or putting them together or trying to work them out? Well, no, because my colleague, Stuart Haslam at Imperial College, and very talented postdocs in his group 20 or so years ago, wrote programs to do the annotations and the drawing of the cartoons automatically, and these are open source informatic tools that you can download from the web or access from the web. So I just want to show you one little bit of MSMS of that neutrophil data, and that's one of the low molecular weight sialyl Lewis X containing components. Most of the sialyl Lewis X on neutrophils is on the polylacnate, so up at the very high molecular weight glycans. But I just want to show you what we can do, even when abundances are low, because of this unique specificity, if you like, of the mass spectrometer. It measures things accurately. This is one little section of that spectrum that I showed you a few seconds ago, spread out, and we're looking at this molecular ion cluster here, and I've spread it out even further here to show you that there is a molecular ion which is sitting, as it were, on the lower mass side of this major component, which is a non-sialylated glycan. And that corresponds in mass to being a sialyl Lewis X containing biantennary disialylated core fucosylated glycan. Now, you might say skeptically, I don't believe it, it looks like a little bit of grass at the bottom. However, if you do MSMS on the overall cluster, then there are beautiful signals, not the most abundant, because it's not the most abundant, but nonetheless, very good signal to noise that tell us unequivocally that we have that component there. So essentially, you are now up to speed with everything we can do with glycomics, and I just want to show you a couple of examples to highlight what we can now do largely from a sensitivity perspective that's so much better than we could do back in the fab days. So if you're thinking about, as you perhaps not, but I am, that very early work on band 3, little I, big I. Little I means linear polylacnac, big I means branched with a lacnac linked to galactose in the linear chain. And in work that we've been doing with Chuck Dimitrov, he has been looking at why or how glycans are changed when melanocytes become cancerous, when they become melanoma cells, and how that affects the malignancy, and in particular, how that affects the ability of the glycans to engage with galactins, in this case, particularly galactin-3. And I just want to show a tiny bit of data, which is on this slide here. The yellow arrows now are highlighting a couple of peaks that I'm going to show you on the next slide. But before I do that, I just want to point out that the normal melanocytes here, if you look at the masses here and the size of the peaks, you can pretty much superimpose the spectra. So the fingerprint is looking identical. There's some changes in abundance for the less abundant high molecular weight components that are there at 2% of the abundance of these. The key thing, though, if we look at those yellow arrowed peaks, and it's true of all of them, but I've just chosen those because they represent extremely well, is that when we do the MSMS experiment, the normal melanocyte pattern is completely different with respect to this big red peak here from the melanoma. And what this means is that this is predominantly linear because it's losing 1 and 2 and 3 lacnacs at a time, whereas when it's branched, it can only lose 1 and then 3 because it has the little branch off the side. And looking at the relative abundance of that diagnostic peak allows us to define semi-quantitatively the little I, big I content of the samples. Another example, which comes actually from Jim's lab, is looking at what happens when people try to manipulate the glycosylation using inhibitors. And glycomics is perfect at showing what might happen when you do that. And I just want to give an example here. This is a metabolic sialyl transferase inhibitor. It has a fucose at this position here. This is the glycomic part of the glycome of the control cells, and this is what happens when you use the inhibitor. So all of the diamonds vanished. That's what's expected. But, of course, what we're seeing is what has happened as a consequence of all of those sialic acids going. And essentially what's happened, lots and lots of fucose on the antenna. Antenna are all growing. So be careful when you use your inhibitors. Don't think, I've got a sialyl transferase inhibitor, so therefore I've got everything the same as it was before. It's just not got sialic acid on. No, it's got potentially a lot of other things happening. And then the final example I want to show is to illustrate glycomics from bands on gels. And this is work of Matt Shoulders at MIT, where he's looking at the unfolded protein response and how that might have an impact on glycosylation. And in particular, this transcription factor here, which is activated when the unfolded protein response is activated under stress. Again, if you want to know the detail of the science, the reference is there. But what I want to show you is just how you can actually take a reporter glycoprotein that is being looked at to see what's happening to the glycosylation and just simply cut the band out of the gel and analyze it. So these are hex cells, actually. So this is the reporter protein. It's a small adhesive protein called CD2. And it separates on the gel, which is quite remarkable, into the small oligomannose glycoforms that are B, and A is slightly more complex, as you can see. When the cells are stressed and this transcription factor is activated, then it's a single band only, and the glycans are much more complicated. And in fact, you can then delve into the kinds of things that are happening with respect to the glycosyl transferases. So the same methodology is used for gel bands as it is used for cells and tissues. I think I'm doing all right, aren't I? Yeah. So I now come to the final, and if I'd run out of time, I would have stopped. But what I want to do, since I do still have time, is to tell you a little bit about work that we're doing at the moment at Imperial College in the March of Dimes. It's a US charity, so Americans in the audience are probably familiar with it, which is a charity that funds research and help to families of premature birth babies, and trying to understand, the aim is to try to understand why there is such a high incidence of premature birth and why the incidence has not improved with improvements in health of the mother. So there's a very, very high level right across the world of premature birth. And it's known that the rates actually vary from country to country. They're known to vary depending on people's blood groups, actually, for example. Okay, so what I want to do, if I can get this to work, yes, is to just give you a few examples of data that we're getting, largely to show you what can be achieved with the methods. So I'm not going to talk anything about the bigger picture of what's being done, other than to highlight that this is a very big, as it were, center at Imperial College that has been funded. And the key PIs are Phil Bennett, Lynn Sykes, and David McIntyre in the medical school and the hospitals at Imperial, where there is a high-risk birth center. And what I mean by that is all women who are diagnosed potentially as being at high risk as a result of what has happened in previous pregnancies or previous miscarriages are referred to this center. So all of the samples that I'll be telling you about are from high-risk women. And the research question that is being addressed is, does colonization of the vagina by pathogenic bacteria lead to preterm birth? And there are some very good and sound reasons why that is a valid question to ask. I just don't have time to talk about the background. I do want to just mention, though, if you're not aware of this, that although it had always been thought until relatively recently that having a microbiome in the female reproductive tract was bad, in other words, it was only because of infections, is absolutely not true. There is a commensal or healthy microbiome that is extremely important for the development and continuation of a healthy pregnancy. And what's very interesting about the microbiome in the reproductive tract compared with the gastrointestinal tract is that a healthy microbiome is very limited in species. Usually there is one dominant species in a healthy microbiome, and it becomes, in the unhealthy microbiome, it becomes a more diverse set of microbes. So the experimental strategy that we're involved with is essentially, here is, 5 minutes left, excellent. Here is everything that you've seen to date. All I want to point out up here with respect to the samples, just so that you know where the samples are coming from, they are soft cup sample collections at the cervix. And what will be in these samples is largely complex mucins. Also, there will be some epithelial cells, lots of neutrophils, and other immune cells, as well as potentially glycoproteins from the serum, IgA, IgG, just so that you have a feel for what's in the samples. And there are two areas of current development of our glycomics that I want to highlight very briefly, so hopefully I won't go too much over the 5 minutes. Our new method development with respect to those overlapping peaks that I exemplified in that neutrophil spectrum when we had those little tiny sialyl Lewis X containing sialylated glycan overlapping with a more abundant non-sialylated glycan, as to what we can now do with those kind of molecular ions. And then I also want to briefly talk about the epitopes that are biologically functional. So the first thing I just want to show, because I think it's pretty, is just a simple comparison of the N-linked glycans of two samples. They're both high risk, but one gave birth preterm, the other gave birth at term. And I particularly want you to notice the difference at the top end here of the polylacnate containing components. This profile here is dramatically different in the two cases. We've now looked at 40 patients with lots of differences, and we're grappling at the moment with what differences might be significant. I'm not going to be talking about that today. But I want to highlight that in other regions of the spectrum here, there's differences in relative abundance, but not significant. So we have the high mannose glycans here and biantennary glycans here. So what about those overlapping peaks? I just want to say that a very talented postdoc in the lab, Gang Wu, has written a beautiful algorithm using R, where he can essentially deconvolute these very complex molecular ions. And the algorithm will work out the intensity, the relative intensity of the different components based on the accurate masses. So in this little bit of the spectrum here, we are seeing 4 glycans. This is a hexose oligomer that almost certainly comes from high levels of hexose oligomers in that cervical vaginal fluid. And basically, the pattern here reproduced by the algorithm, and these are the relative abundances. I put this slide in at the last minute because I suddenly remembered there are a lot of mucin people in the audience, and I hadn't said anything about the O-glycans. So the only thing that's important is the bit at the bottom. Essentially, the O-glycans, we analyze exactly the same, and we get lovely patterns. They are smaller, but it's the same principle. Last few slides, hopefully we'll not go over that last couple of minutes. How do we define all these terminal epitopes that are so important biologically? Well, this is what I've already talked to you about. I showed it with the neutrophils with the sialyl Lewis X. What we can also do, if we're looking at the polylacnate components, and this is incredibly powerful for these kind of analyses, is to use an enzyme that everyone has used for 30, 40 years, endo-beta-galactosidase. Absolutely lovely enzyme because it cuts those linear polylacnate chains. And therefore, you can see all of the non-reducing ends of the polylacnate chains. And this we're calling the glycan epitome. And basically, I hope you can see this here. If, depending on where the modification is, the enzyme won't cleave. So that's why we get the longer epitopes here. And we can ask ourselves questions about the level of fucosylation as to how much blood group is there. Is the blood group in tandem with other epitopes, like, for example, blood group A here in tandem with a Lewis structure. All of these structures are present in abundance. And I wanted to finish with Ku, because Ku did his PhD with me, has gone on to a stellar career here, has worked so hard to organize this meeting, but is also able to do wonderful science at the same time. And Ku has taken some of our methylated samples, the ones that I already showed you data on, and he's using his Orbitrap here to look at the fragment ions and then to fragment them again, which is MS3 for those who are new to mass spectrometry. And what he can do with this MS2, MS3 is he can then quantitate all of the individual isomeric forms of the compositions that I was showing you on the earlier slides. So Lewis A versus Lewis X, for example, which, if you're familiar with this, you'll know what I'm talking about. So Ku has promised that if you have any questions on this, he will answer them in the question time. Some of these observations are in this paper. And just to finish, I just want to say that my colleagues, Daniel and Manfred, are going to highlight a bit more of the yellow-colored bits and everything else that's on here, plus extras in their talks. And thank you very much for your attention.
Thank you, Anne. So we have plenty of time, 10 minutes. schedule for questions and answer. Anyone wants to start off with? Sure.
And thank you for a very informative talk. You mentioned that this have a tag at the reducing end, or sometimes people do the reduction, is very important because that enable you to tell you which size the reducing end, which size non-reducing end. But how about this non-reducing oligosaccharide, which exist in abundant in plant? Some of these oligosaccharides are non-reducing oligosaccharide.
So that you can't tag them, you're saying?
So
Yeah. Well, yes. I mean, if you can tag, the permethylated samples are just tagged with having a methyl group on, to be honest. So if they don't have if you can't put an additional substituent on the reducing end, that's not a problem because you can interpret from the overall, as long as you fully methylate or fully acetylate, which can be another way of doing it, particularly if you're looking at bacterial oligosaccharides, it can be useful to acetylate. So it's not mandatory, it's just that it's extremely helpful because it's not only differentiating, it also directs the fragmentation. So you get different fragment ions that are the reducing end fragment ions. Okay. So you just simply tailor your experiment to the sample you have. Does that answer your question? So ones that are non-reducing work beautifully. Are you thinking about things like tree halos, for example, at the reducing end? Yeah. Yeah. Yeah.
Are there any glycan modifications that permethylation can remove and obscure?
That's a very good question. So yes, there are. Anything which is labile to base will be removed by the permethylation. So if you have, for example, an O-acetyl group, and if you're working with sialic acid, you might be particularly interested in that, then you have to tailor your experiment to look for the labile groups. So permethylation, you would do your permethylation just to get your overall picture. And then, for example, you might want to do, you could try it native, but if that's not sensitive enough, you could then acetylate using conditions that don't remove natural acetates, and you use a deuterated reagent. So we, and others as well, over the years, have looked for modifications that are labile by using, for example, deuteroacetylation or equivalent procedures.
Interesting. You saw the difference between term and the preterm glycan patterns. Is there a difference between the gestational age of your so-called preterm?
That question has a multitude of answers, I'm afraid. I'm not trying to be cagey, but I just need to say what we are seeing generally. So at the moment, we have about 40 individual women. And amongst those women, we have the 3 blood groups. And we also have different ethnicities, which we know affects term and preterm. And we also have to remember that all of these women are high risk, even if they did go to term. And some of them have gone to term because they've had interventions. And I don't know if you're familiar with interventions like cerclage, and they've had progesterone sometimes. So what we're trying to do at the moment is to unravel all of these extra complications because we don't have a control term with a control preterm. What we've tried to do is to say this is a term that's blood group A and this is a preterm that's blood group A. At least that's one comparison. However, as it turns out, there are very, very few preterm A's. So our current thinking is that having blood group A is somehow protective in high-risk people. We don't know why. So we don't really have an answer to your question at the moment. We have some really, really interesting data and interesting differences. but we don't know whether that maps onto term versus preterm or on what the actual individual woman has had as treatment throughout her pregnancy.
But what are the gestational age for your preterm in most cases?
The earlier, well, okay, so we have gestation, the earliest is 25 weeks. And the latest is the definition, which I didn't give actually, which you might be aware of. Preterm is regarded as any birth less than 37 weeks. So we do have some that are very close to term, but the majority of our preterm are 25 to 27 weeks. Another little interesting comment before I go to the next question, by the way, is our work has come to a grinding halt at the moment. Well, not entirely grinding halt, because during COVID, there were virtually no preterm births during COVID in our high-risk centre, which everyone is thinking hard about at the moment. It means that we haven't got any samples. Sorry, there was a question up there.
Thank you for the talk. So you mentioned in the cervical vaginal fluid, there are different cell types. So when you see the glycan, the differences in the glycan structure, is it on a specific cell type? Do you have to isolate them before doing the mass spec?
Right, so what we have done is we've separated cells away from other stuff. And that was at the beginning of that slide. We have raw material, the more fluidic. With respect to the cells, it's dependent on the status of the woman, actually, as to whether there's lots of neutrophils. And you can actually measure the neutrophils, so you know if it's a high cell count. The really great thing about neutrophils, which was exemplified in the data I showed you, is that we know exactly what the profile is, and it doesn't seem to change in pregnancy. So we can use the neutrophil glycome pattern to say those are the neutrophil glycans at high mass. And then we can start to add on to that what might be the epithelial cells. So we're not at the moment trying to separate, as it were, in the lab, but we're separating by using our knowledge of the glycomes to look at the data and actually another algorithm that Gang Wu has written is to pull out all the neutrophil glycans out of the data, and then we have the data remaining without the neutrophil, because they're the main cell, as it were, component. You might call it contaminant, but it probably isn't. Neutrophils are probably there in most cases to help rather than to hinder, we think. But it's hard to know at the moment.
Any more questions? I should just add one comment about the question that I think for permethylation also that the phosphate and sulfate will stay on. And then
yes,
also that if you look at lower organism where you have O-methylation, so one of the techniques people can use is deuteropermethylation to distinguish between the natural metal and the deuterometal.
Yeah, I should certainly have mentioned that because Ku has done some lovely work on sulfur glycomics, optimizing the sensitivity for sulfur glycomics. Yep.
Thank you for your information. Can you comment on how important for quantification of ROSA fragment? Because in the signal of the MS, actually it's really not really reflected the really quantity of the species of the glycan itself.
Yeah, so that is actually also an important question. It is true that the abundance of a peak will not directly map onto its abundance because obviously there are factors relating to the ability to ionize, etc. However, once permethylated, all of the complex glycans have a very similar level of hydrophobicity. High mannose glycans tend to be a bit different. Their abundance will not directly correlate with the complex. So the short answer to your question is once methylated, comparative quantitation is really accurate. If you're looking at a comparison of a set of glycans in one spectrum compared with another, completely reproducible and very, very accurate. But be careful comparing between classes. But if it's native, you're absolutely right. quantitation is totally meaningless. It's a bit like proteomics, where quantitation of peptides is meaningless unless you have standards for everything. Okay, thank you.
Okay, I think in order to keep time, so we'll ask the next speaker. And the next speaker is Daniel Coleridge. And while he's switching over the computer, I'll give a brief introduction about Daniel's career. Daniel came from Austria, right? And then mostly he got his Doctor of Science in Austria, and then he did a postdoc, and then most importantly, he also went to Macquarie University in Australia to do a postdoc there, I think with Nikki Packer. And as you know, in the field, Macquarie University is one of the important centers for glycomics and glycoproteomics even today. And then he moves to Potsdam to be associated with Max Planck Institute of Colloids and Interfaces, and stayed there until 2017. Before, I think he likes Australia very much, so he went back to Australia, and now he's in this Institute for Glycomics in Griffith, taking up the leadership for this cancer glycomics and glycoproteomics initiative at the Australian Centre for Central Glycomics. What you call that A2CG? What do you call A2?
Australian Centre for Cancer Glycomics. So, to be honest, I don't remember anymore.
Okay.
You got me off guard now.
Okay. So Daniel is very famous for everything, and including the law. You probably have read the Nature Protocol articles, and where they developed this method of PGC and this blotting to VDF, etc. And he's working on these glycoproteomics, and he is also one of the person that devoted a lot of effort to the Mirage. Are you going to say anything about Mirage?
Yes.
Okay.
I'm going to mention it.
Okay, so all time to you, Daniel.
Okay, here we are. Perfect. Thank you very much, Ku, for this very great organizing this meeting here. I think I can already see it's going very, very well. Everything is working smoothly. And what I'm going to talk today is more about, my aspects of how glycoproteomics, or the considerations in glycoproteomics that we need to be aware of, and also making you aware of the fact that you're not going to solve everything the way that you want to solve it. Yeah? So be aware of the fact that sometimes you will not be able to answer all the questions that you would like to answer from your spectra. So let's get started with some of the basic considerations. First of all, why are we looking into the glycocode? And that's the beauty of this conference. I don't have to explain too much about that. But one particular aspect that I want to point out is that while especially proteomics people tend to look at this very linear way of the genome, essentially defines a transcriptome, and that essentially defines the proteome in one or the other way. If you're starting looking at the glycosylation on there are a lot of other factors that in the end of the day will lead to the way how the glycoproteome, how the glycans are actually shaped on a cell. And external signals will impact how the metabolome will look like, will impact how essentially the rest in there looks like. And that's where glycosylation comes into place, because glycans are being influenced by all of the different factors, and the way how the glycoproteome will be shaped in the end of the day will be a consequence of these, in addition to the genetic, transcriptomic, and other factors that we have there. The other part that we need to think about when we want to look at glycoproteins is we really need to think about, how are the glycans attached to? And there are so many different types of glycans, and Amber already provided a great overview of what glycomics is looking usually in terms of O glycans and N glycans. There are, of course, glycolipids as well that are quite important, but if we're looking at glycoproteomics nowadays, we are pretty much focusing on N glycans and O glycans that are quite traditional. We hardly look at GAGs or any of the other really, really interesting glycans that are out there. And we differentiate them by what they're attached to, whether they're N and O linked, and there's so many different versions of that, but also essentially there's a difference of what is the first sugar attachment. Yeah, N glycans and O glycans are the ones that everybody thinks of, but there is O fucose ones, O mannose ones, O glucnag ones, which are different type of glycoproteins, but glycoproteins nonetheless. So depending on where you're coming from, what your particular question is, you're going to have a lot of different opportunities to already decide in which direction you want to go and which direction you need to go for your project. If we need, and if we want to understand glycoproteins, we need to think about a lot of different aspects. We want to identify the protein, ideally also want to quantitate the protein, and that can very often be done purely on non-glycosylated peptides as well. We want to know the size of glycosylation, which for N-glycans is usually not the biggest question because we have an N-glycosylation motif that we, in most cases, is just present as one in one particular peptide. But very often we might want to know the site occupancy as well. This is something that is not talked about very often. To which extent is a site actually glycosylated? 50%, 90%, 100%? What are the types of glycan present on a specific site? So the heterogeneity, the microheterogeneity on a specific site is important. And what's the quantitative distribution? A lot of the glycoproteomics tools nowadays give you an ID. They identify the specific type of glycopeptide, but actually not a lot of them really give you an overview what is essentially the major type of glycosylation present to 80%, to 70%, to 1%. And this is something that can be actually quite interesting to look at what's the quantitation difference of glycosylation structures or compositions on a specific site. And also, most but not least, in glycoproteomics, in most cases, you will just reliably able to say a composition rather than a structure. And this is something that I see a lot in reviewing papers, that people very likely overinterpret the spectrum or put structures onto the glycopeptides without having any additional support that actually gives them an idea whether this is a 2,3 linkage to 6 linkage of a sialic acid, whether this is essentially core fucose or whether this is a Lewis fucose. And be mindful, there is essentially, especially in positive mode, a lot of opportunity for Lewis fucoses to pretend to be something else. Okay, and of course, in the end of the day, we want to know this not just from a single protein, but we would like to know this in a system-wide context. As important as it is to know this for single glycoproteins, in the glycoproteomics experiment, you usually would like to look at it in the whole context of the cell. And these are, especially these three parts essentially, are the ones that are very specific for glycoproteomics and that proteomics people usually don't have to think about. One other aspect that we need to think about is actually the fact that the variation, the number of unknown factors that come into place are so much higher in doing glycomics and glycoproteomics compared to proteomics. First of all, we have a very direct translation between the nucleotide sequence and the protein sequence. So proteomics wouldn't be there where it is today if essentially proteomics people would have to de novo sequence each and every peptide. The fact that we have the genome sequence available makes proteomics actually possible the way that we know it nowadays. We don't have quite that luxury in glycomics. Also, despite the fact that in theory a hexamer of sugars could have 192 billion type of combinations, nature has been kind to glycoproteomics and glycomics people because at least in mammalian cells the number of possibilities are much, much reduced due to the fact that we know our biosynthetic pathways and based to the fact that they're very, very well established and conserved biosynthetic pathways for mammalian cells. I wouldn't dare saying this about bacteria anytime. But nonetheless, if we put this together now from a sugar and peptide perspective, thinking about the different variations and opportunities that you would have in terms of sequences, just looking at the hexamer of peptide and the hexamer of a sugar component on there, you have a lot of opportunities what combination that could be. And while this sounds now like a completely panic attack motive approach on there, the Hitchhiker's Guide, so to say, tells you don't panic. That's the most important part that actually tell you, and that's the same that you should actually apply in the context of glycoproteomics. Just because it's possible doesn't mean it's actually there. And also the fact that nature has been able to actually use glycans in such a way to make so many more biomolecules out of a comparable simple set of potential proteins that we have on there is actually the opportunity that we have in glycobiology to understand where the glycocode actually comes into place in defining cellular life. And of course, if we start thinking about the different extensions that the sugars are going to have on there, that's where the challenge comes into defining that. Also, just a reminder there, the fact that this is a non-template biosynthesis, and it's always something that you have to explain to non-glyco people, that there is no way we can actually predict how it's going to be. We have biosynthetic pathways that allow us to say it, but we need to actually really look at the glycans in the end of the day to understand what is happening in there. And in that way, glycosylation for me is essentially my inverse way of playing Lego in one or the other way. I see that nature's way of having a very distinct set of building blocks, but out of these building blocks being able to do two very different things. You take the same building blocks, but depending on how you put them together, you get functionally very, very different aspects. And what we're doing in glycoproteomics and glycomics in one or the other way is taking these things apart and trying to understand what has actually nature been using to put these things together. Why this is important, and this is just some two very, very, very well-known examples to a certain extent, is why we want to know this in a protein-specific context. Is if you're looking at IgG, for example, the way how glycosylation regulates IgG function with respect to ADCC interaction and with respect to our understanding of the pro- and anti-inflammatory features of immunoglobulins is completely independent from the antigen binding. So if you think about the function of glycosylation for IgG with respect to antigen binding, you're not going to find anything. If you think about it with respect to what happens biologically with the molecule, there's a lot of different other functions where glycosylation is relatively important in actually organizing this molecule. On another example that came out of Nikki's lab not such a long time ago, they looked at CD52 on there and found that just a very distinct subset of the various many glycoforms that CD52 comes in is actually biologically active, at least biologically active in the assay that they have been using. That pulls up a lot of questions. Is it essentially just these particular ones that are really important, or are there other biological activities of CD52 where the other forms of glycosylation might be important? Maybe it's just not captured by this particular assay. Nonetheless, you need to have essentially first the tools in place to capture and characterize the heterogeneity, the type of glycans that are present on this protein to ask the biological questions where glycosylation plays a role in here. And the other part where, of course, glycosylation is interesting and relevant for us is that the same protein sequence can actually be very differently glycosylated in different organs. And this is just an example of carcinomeric antigen from our lab, looking at the colon adenocarcinoma one as well as the liver metastasis adenocarcinoma one. So depending on where in the body this protein is being expressed, it exhibits a very different form of glycosylation, which of course increases the complexity of how you have to address it from a glycoproteomics perspective. So, if we really want to understand now glycoproteins, we need to look at them essentially from a more holistic point of view, trying to understand structure and trying to understand the glycosylation and protein sequence at the same point. And we want to capture the whole biology around that. So how can we approach this from a very, very naive point of view in the sense of, okay, what are the key factors that you need to think about when you start out for your experiment? First of all, there is not a single method that's going to give you all the information. So you have to think about what do you actually want to get out of your experiment. And that essentially factors like what amount of material do I have available? Do I have cell lines or do I have to work with tissues? What instrumentation do I have available? And what do I want to know? Not for every biologically relevant question, you need to go essentially with a Ferrari of methodologies. Sometimes the VW Golf tells you pretty much all what you need to do. So just think about your horses for courses in that context, and there's not a perfect way of doing things. If you now want to sequence the glycoproteome, depending on the questions that we're asking, we can go different approaches. Of course, if you're looking at the protein-focused approach, that's how we like to call it on there, where we use the proteins, deglycosylate them with PNGSF. Looking at the deglycosylated peptides, we can still get a lot of information that is comparably easy to acquire and that is relevant with understanding about occupancy levels, but also which sites of glycosylation, at least for N-glycans, have been glycosylated at some point in time. And that might be also from a quantitative perspective all what you might want to know for your specific experiment. On the other hand, of course, if you're looking at glycomics, it's essentially the inverse part of that. We're looking at the sugars, and we do this all very much because we want to understand the details of the structures that we sometimes can't get in any other way, but we also lose essentially the information of which sugars have been attached to which protein unless you're working on a very purified protein, which is where glycoproteomics comes into place. So in a glycoproteomic experiment, you usually end up having 5 major points that you need to think about. First of all, how do you get your protein from your sample of interest, whether this is a cell line, whether this is a body fluid, whether this is a tissue in one or the other way. How do you get your protein into a way that you can work with it, the extraction process. The second part is, okay, how do you fractionate your samples? What do you do in order to get your glycopeptides, your glycoproteins in one or the other way separated? Do you use healing enrichment? Do you use another form of enrichment? Do you digest first before you do enrichment? Do you digest later? So there are a lot of different combinations that you can apply in this particular part before you get actually to the cool part about actually using the mass spec finally and acquiring the data. But even there are a lot of opportunities and a lot of different ways of how this can be done, also different technologies that are out there. And then once you think you've done it, actually the work starts because you need to actually analyze the data. You need to look at the data, and you need to make sense out of that, and that's a question for itself. But it's also important in the end of the day, and I'll put this out specifically, to have good reporting practices. This is something that is becoming more and more relevant and more accepted, luckily. But I'm pretty sure everybody of you had at some point the experience that they were looking at the paper and were just wondering what enzyme did they use or how did they actually do it, because it's not properly reported in there. But if you think about it from that perspective, all the data that we are actually acquiring nowadays might be amazing, might be great, but in 10 years' time, somebody that might actually be able to use this data for a completely different question, whether it's a bioinformatics person that wants to develop a new algorithm, can't actually use this data properly if they don't have all the information properly reported in the first place. So it's not just something that is important for the paper itself, but it's something that, in my opinion at least, is really important for having your research available for other people to use as well. And that's what I think research should also be about. Okay, looking at stage 1, we are picking this out a little bit on there. And this is a poster that you see from one of my students later this week, where we just tried a very simple question: is how different do we get our results depending on the type of extraction methodology that we use. So we keep everything the same, but we just use different technologies or different ways of extracting the proteome. And not really a big surprise, but why we were interested in that, and you see it's very different depending on the type of extraction technology that you use. One that is more specific for membrane proteins, one that is more specific for cytosolic ones. You're going to get enrichments for different aspects of different proteins on there. But the reason why we were also interested in that is depending on how you use your extraction technology on there, you're going to get a different glycome as well. Not like the structures are going to be completely different, but the type of intensities that you get for the different structures will be slightly different, at least in our case. So it's just you can mess up or you can make a difference between your paper A and paper B just based by the fact that the extraction methodology that has been used, even if it's the same cell line, even if it's the same type of body tissue in one or the other way, has been different. And if you want to know more about this, see essentially Pauline at the poster. For the second part on there, and I'm just going to bring some examples about how you can use protease and glycosidase digestions in a way to actually get you more information on your sample. So in the majority of cases, most glycoproteomics experiments would use a type of bottom-up approach. You would digest your protein, and Manfred is going to talk more about intact mass part later on, but for most applications, especially for complex mixtures, the bottom-up approach would be pretty much the way how people would go for it. If you use that and you use enzymes like trypsin, for example, glycosylation can actually impact the way how your enzyme can actually access the certain sites. And this is something we published a couple of years ago where we looked at secretory IgA. You can see that pretty much all of the glycans that carry core fucose were majorly on the peptide that has one miscleavage. And the glycosylation site, the N here, is quite close to this, to the arginine that is being cleaved by trypsin on there. So in one or the other way, the presence of core fucose definitely impeded, to a certain extent, the ability of trypsin to cleave appropriately on this particular site. But you can see that you get a very different quantitation of the different glycoforms depending on the type of peptide that you actually look at. So glycosylation can impact the way how your protease of choice is actually going to work. Other proteases that are now becoming more and more relevant are actually mucinases. And these are amazing enzymes that actually are able to cleave now in mucin type of domains that by trypsin or most of the usually used proteases are actually quite difficult to get to because they have a lot of prolines in there, a lot of cereins and threonines, hardly any of the normal classical enzymes that are being cleaved. So if you're after O-glycans, if you're really interested in the mucin-rich domains, mucinases are the enzymes that you would want to actually use for this particular purpose. There are a lot of different ones out there. They have quite a great specificity towards also the sugar part sometimes and the type of amino acid cleavage that they're actually linking or cleaving. But yeah, that would be the ones to go for mucins. N-glycan release or releasing glycans essentially in a different way can also be something that you can use to get some glycoproteomics information out of that. So we know we have PNGAS-A or PNGAS-F that can actually cleave glycans, especially if you're in the insect field or plant field, you would want to use PNGAS-A because PNGAS-F is unable to cleave any core alpha-1,3 type fucoses. So by removing an N-glycan, you're going to change the asparagine into aspartic acid, and that can be used to identify the site of glycosylation. However, using different enzymes like endo-H enzymes, for example, that cleave between the innermost glucnac and the rest of the glycan on there, you can retain some information of the glycosylation on there, but you can also use the specificity of the endo enzymes in order to get some information about what type of composition or structure has been attached on this particular site beforehand. And this has been very neatly done by Jim's group essentially a couple of years ago, where they used the combination treatment of endo-H and PNGAS-F with O18 labeling on there to differentiate and easily quantitate between complex oligomannose and non-glycosylated sites of glycosylation on influenza viral membrane proteins on there. So that's a very, very neat and easy way to do this. Also not very complex. They were not interested in this part about what specific are the glycosylation forms of all of these sites. They wanted to know and were interested in complex oligomannose versus unglycosylated. And that's a very, very neat way to actually look at that. And you can see how the different sites quantitatively have been differing on there. And the reason why they could actually do this also quantitatively is because we were able to show a couple of years before that the glucnac itself doesn't really impede with the ionization efficiency of the glycopeptide compared to the larger structures in one or the other way. Okay, so enrichment and fractionation is something that in a lot of glycoproteomics experiments is used very widely. So I'm pretty sure all of you have heard about lectin chromatography at some point, about hydrosin technology, about HeLiC in one or the other way. All good and all very, very important, but none of them is perfect. So that's why I'm purposely saying enrichment, because all what you're doing is essentially fractionating and enriching, but you're not isolating glycopeptides. Because isolation, per my definition, would mean that all of them are being captured, and this is something that, in my opinion, is not something that can be achieved. Depending on what you actually do, there's a great review from Nick Riley out there that really summarized all the different type of glycopeptide enrichment technologies out there. They all employ different type of chemistries. They either use the hydrophilicity of the glycans, or they use certain features like the charge from the sialic acids for the enrichment, like titanium dioxide, for example, does. PGC uses essentially also the hydrophilicity and in enriching glycopeptides and glycoproteins. And glycopeptides in particular on there. But then there are the chemical coupling technologies using essentially sugars that are being embedded within the glycan that then chemically can be pulled out, or other type of tools. So look, in terms of enrichment technologies, I can really highly recommend this particular review. Select the ones that is most suitable for your particular project, but be aware that it's always going to be essentially a compromise in terms of enrichment that you have to do. Okay, if we're now looking at the most important part, the data generation part, because without that, in the end of the day, you can't do a lot. There are different ways of how glycopeptides can be analyzed. And of course, the classical CID-based dissociation will not give you a lot of peptide information. It usually would give you a lot of glycosidic fragmentation from the glycan on there, but you won't get a lot of peptide information. That's where HCD comes into place from the collisional dissociation methods on there that really cleaves off a lot and it gives a lot of energy into the peptide, which usually allows you to get a very good idea about the peptide sequence as well. But you're going to be limited in terms of seeing a lot of information from the glycan. So kind of sequencing or getting an idea about some of the glycan compositions on there, apart from the calculating composition point of view, will not be really possible. ETD-based fragmentation technologies will remain the glycan intact, but will essentially really allow you to fragment the peptide backbone in a very nice way, and that's a very good way of getting much more site-specific information or site information, especially for non-N-glycopeptides. But usually works very well on highly charged peptides. So doubly charged glycopeptides, if you see them, would hardly ever fragment very well in ETD and give you a lot of information there. And then there are, of course, the supplement, the kind of hybrid type of technologies that use ETD as well as a little bit of collision dissociation in there to give you more fragmentation information there. So there's a lot of different fragmentation techniques out there, and why I'm talking about them is that for glycopeptides, optimizing them is something that is not that easy, because there's a lot of factors that influence the way how glycopeptides are going to be fragmented. Usually glycans would fragment off first, and we did this a couple of years ago, where we actually looked at the optimal fragmentation for the different charge stages, and then you can see that every charge state has quite an optimal fragmentation energy that is required to order get actually a good coverage of the peptide backbone, because in the end of the day you need to understand the peptide backbone in order to be able to assign it to a specific protein. And of course we can use a lot of the different diagnostic ions to actually get a lot of information. So identifying a glycopeptide or as a glycopeptide is usually not that difficult, just based on the oxonium ions that you get in the glycopeptide spectrum on there. But getting essentially a good oxonium ion series as well as a good Y and B ion series of the peptide backbone, as well as a good capital Y ion series, is something that is requiring a good tuning of the instrument and optimal conditions. And why I'm mentioning is that important to actually not be aware of what can happen within the gas phase. There's a lot of things happen, and that can provide you with fragments that pretend to be something that they're actually not. And and it's nice to see that acetylation and methylation of glycans can actually also help us in glycopeptides. And here in that case, that was actually a natural form of methylation, acetylation on a flagellin from a bacterial species on there, where we did the Glycoproteomics characterization. And when we looked at that, and that was done on an ion trap at that point in time, when we looked at the fragment spectrum on there, what you could see is essentially that we got nice glycosidic fragments essentially down there, and we did a separate Glycomics experiment on there to understand that this is actually the sugar and the arabinose that is linked to a glucnec that is present on this flagellin. But when we looked at the fragmentation, we saw 2 Y1I fragments, one that indicated that the fucose is attached to the peptide, and one which indicated that the glucnec is attached to the peptide. So based on that, it was a little bit difficult to actually understand what's going on here, because on the Glycomics part we didn't see that. Luckily, this bacteria also had essentially methylated arabinose as well as acetylated arabinose on there. And what we could really very nicely see is that as soon as it was acetylated, there was essentially hardly any gas phase migration present of this arabinose onto the peptide. And that's something that has been reported earlier as well, that acetylation actually pretty much abolishes this type of gas phase migration that you can see in the positive mode, in particular there. And given the fact that also these fragments essentially started increasing in this particular way as well, we were quite confident that this actually then corresponds to what we saw on the Glycomics part. But the point why I brought this up in here is especially the oxyhexoses seem to have this tendency to migrate around in the gas phase and can give you sometimes a rise of peaks and fragment ions that you could actually assign in the wrong way. Okay, which brings us to the point of data analysis. And there are a lot of different ways of how it can be done. Mind you, as I mentioned already in the beginning, sometimes your data will not be able to tell you everything that you would want to have out of it. Yeah. So in that case, I would really say, rather than over-interpreting it, I would highly recommend to essentially stick to the data that you have. Do orthogonal, if you have the ability, orthogonal ways of identifying structures, identifying sites of glycosylation, depending on what the question of your project is. But try to not over-interpret it, because that can go very, very wrong very quickly. There are a lot of different glycoproteomics softwares out there, and the last couple of years has seen a lot of development in the glycoproteomics software development. I still, maybe I'm a little bit overcritical on there. I want to see a little bit more data coming out of them, especially being used by non-developers, but actually by people in the field on there, to actually believe much more how reliable they actually are. And there was a study that was led by Morton's team out of Macquarie that you might have come across in one or the other way, where we looked actually at how does the interpretation of glycoproteomics data look like in the different labs. And for that, 2 raw files of glycoproteomics data that have been acquired were sent out to people, and they were just supposed to analyze the data using their bioinformatics software of choice, which for a lot of the user parts was essentially Bionic. There was the developer part where different people used their particular software that they have been developing. And then the data was compared, and the outcomes were essentially compared of the different softwares and of the different participants on there. And the interesting part is that there were a lot of, and I'm sorry for this quality of this, that should be better. It looked better yesterday when I put this again in. So what you can see is essentially it was all the very same data file, but there was quite a difference in terms of how many glycopeptides and what type of glycopeptides have been reported across the different groups. And while I'm not going to go into too much detail with the paper, if you have a chance on there, there has been a comparably low number of very consensus glycopeptides that have been essentially identified across all groups, and there was a huge number of glycopeptides that have been identified by just a single group in one or the other way, which turns out that in the end of the day, there's some groups that did a little bit of data let's say, revision in the sense of actually looking at the data and not essentially accepting every glycopeptide that has been actually put out as a potential glycopeptide, and some that essentially did a little bit less work on it. However, Rebecca and Morton were then able to kind of use this data also to kind of devise a strategy, and that was just using Bionic, as it was the one that was mostly used by the users, to identify what would be the most suitable settings to give you a most reliable information out of there, but that are also usable in the terms of having a data analysis time of not 3 days in order to get your sample analyzed. That's all in the paper on there if you're more interested into that, but there are a couple of key components that essentially made quite a difference in the quality of the data that you could actually use, and one of them is also really defining the glycospace. So if you have an opportunity to define the glycospace, but not just adding each and every glycan that could be possible, but actually adjusting this to the type of glycosylation that you have present in this type of tissue, in this type of body fluid, in a more educated way, you can actually already improve your outcomes tremendously. That brings me shortly to mentioning the Mirage initiative. So the Mirage initiative has initially been done for Glycomics, but essentially includes all type of glycoconjugates, so to say, and also all aspects of the way of how we look at glycans. And it's really supposed to be a guideline and a support for researchers to give you an idea what are the critical parameters that you should essentially include in reporting your experiment. Exactly what I mentioned beforehand already. So have a look at that. There's a lot of work going into the Mirage one by a lot of people around the world, which is supported by the Balchner Institute. There are a lot of other glyco resources, and many of them are here. And there's a bioinformatics session later this week as well, led by Kyoko on there. So there are a lot of tools that can help you in different ways of getting information that's already out there, having different tools on there. I can just highly recommend to go and look at these at the informatics workshop tomorrow. So just now to sum this shortly up, look at these different ones. These are, from my point of view, the 5 most important aspects within an experiment that you need to look at, that you need to think about how are the best suited for your particular problem. And the way how I see it, glycoproteomics is kind of the centerpiece of the puzzle. If all of the different omics technologies are out there, that in the end of the day will help us to really understand what is happening within the cell. Let's finish this off with a quick example that kind of shows what you can do with this type of technologies and or what information you can get out. And that's an example where we looked at environment-mediated drug resistance in the leukemia cell line model. And the reason why we wanted to look at that is really to look at the first stages of acquired resistance and what is happening on the glycoprotein on there. That was work that we did together with a group of Nora Heisterkamp from City of Hope, where we looked at different levels of incretin exposure on these co-cultured B cells. It did RNA sequencing, did high pH fractionation of the proteome, we did Glycomics on that, and also looked at the glycoproteome and proteome there. And that was something that Thiago was leading, one of my PhD students that's now actually in Vienna. When looking at the transcript and proteome part on there, you can see all the usual differences. I'm not going too much into the detail of that because there's a couple of logical things and a couple of things that were unexpected. But we were more interested in the glycosylation aspect. So we looked at the glycosylation. You could see there was not a lot of change depending on the type of exposure to incretin. Some slight changes maybe within the N-glycans, but yeah, not too much. Sorry for that, except of the fact that MGAP3 was the only one where we could see both on transcript level, but also on the level of glycosylation, an increase of bisected glycans in there. We then started looking at the glycoproteome in more particular detail. I'm just talking about CD38 because overall there was not a lot of change, but there were a couple of proteins where the glycosylation changed specifically. And CD38 was an interesting one in here because it's an important immune cell receptor involved in cell adhesion, but also associated with the pathogenesis of leukemia in general, and also immunotherapy target in already multiple myeloma and also investigated in other forms of leukemia. So while it didn't really change in the transcript and protein level on there, when we started looking at the glycosylation, at the site-specific glycosylation, quantify what type of glycans are actually there, we could see that in the resistant cells, we got an increase in oligomannose-type glycans, so from less complex to more oligomannose, on asparagine 100, as well as on asparagine 219. But so it's not a complete change, not a complete shift, but it's essentially a shift that we were able to see based on the quantitation of the glycopeptides. And we're now starting to look at the function-structure relationship of CD38, and it appears like that the fact that there's more oligomannose-type glycans present on the sites is essentially a consequence of a rearrangement of the CD38 complex on the membrane that actually changes the accessibility for the different exoglycosidases or mannosidases to then actually convert it into a complex type of glycans. So it's kind of an indication, this glycoproteomics data gave us an indication about a change in the oligomer formation of CD38, which we're currently trying to confirm. Okay, with this, I'm finishing up on that. I want to say thank you to all the amazing people in my group that actually do a lot of work. You for your attention, and also our different collaboration partners and funding agencies. And this is not a full list. There's many more in there. Yeah, thank you very much.
Thank you, Daniel. Again, we have 10 minutes for discussion. And I'm sure we have plenty of questions to ask.
Yeah. Hans.
Working that. So what's about all these different softwares? I have a question. I see a lot of papers where they compare or claim their software is better and they only have numbers. But what I would like to see, show me a spectrum and tell me why is this one finding this one and the other one not. So
I completely agree, and that's the problem that we have, that some of them are published, but you can't actually access them. Because they are still a code somewhere, or they're not usable for somebody that's not a software developer. Yeah. So while I think there's a lot of new tools coming out, a lot of new algorithms being developed, they're not all there yet that you can actually use them. and actually use them as a non-professional program developer on there. But also I think the benchmarking of these is something that at least this data now that we have from the study that Morten led there allows us now to benchmark new algorithms and at least provide them with, if they actually recognize this accurately on there, because there has been a lot of manual work going into the identification of a lot of the spectra in there. So that's where this software and these data sets can actually also be of great support. Yeah. I hope that answers the question.
Not really, but I see.
I
see the problem indeed, because I don't see why one program is getting more than the other, or they sort of claim they have their algorithm is better and they describe it more or less, but
they use different signals. They use sometimes signals that, in my point of view, I wouldn't use because they're kind of
yes.
just there. But that's the part
because
where
like the 204 mass for the Gluckner, my experience, you have it in half of your spectra, even in the ones that are not glycopeptides. Maybe they are somewhere in the background, but so I would not use that at all. Or you should have it, but
I would use it not as the only one. You usually, if you have a glycopeptide, you would have more than 2 or 4. You would have more than
you need more, of course.
Yeah, there are more than different options, but you don't always get all this information, especially from a new development, and especially if there might be commercial interest behind in this development, where they try to publish but don't tell you everything. So that's a little bit the
so it's a black box. You have to. That's the problem.
Yeah, I agree.
Thank you very much, Daniel. So I love you brought up the mucinases for the old glycoproteomics. One thing we should all consider is that not only do the mucinases have a peptide substrate specificity that's a little wacky, but when it comes to the glycan requirement, they will have very different requirements, and so efficiency when it's like TN, or some sialic acid. So your data will be tremendously biased. I spent yesterday night reading a new paper that came from Cummings Group with a fantastic brain glycoproteome. And there, they suddenly come out with the conclusion that surprisingly there's a lot of TN glycopeptides. Now, if your mucinase selectively cleaves the TN, and you're using total cell lysates, you're enriching for intracellular intermediate byproducts.
Yep. And thank you very much for actually pointing this out, because this is exactly, it's amazing to have these mucinases. There are a lot of stuff that we still need to understand on the mucinases, but they are still better than having nothing in order to actually cleave in mucin domains or starting to analyze mucin domains at all. So it's just always a compromise, whatever we're doing. But that's why it's also important to really go back in the literature and go back to what these tools actually can do and not overinterpret your data. So yeah, thank you very much. And pardon? And of course, doing the glycomics aspect, as I mentioned, it helps. Why not doing it? Because we can do it, and you get 2 orthogonal data sets that have to make sense. Yeah.
Thank you so much for covering a very broad spectrum of approaches. We spoke a little bit about this over breakfast today, but I thought it might be interesting for the audience if you wanted to touch on the potential of ion mobility and collisional cross-sectional data to move from actual compositional to structural resolution, stereostructural resolution.
It's a very good point, and I think ion mobility offers a lot of opportunities in there. I don't think we are there yet to use ion mobility in a way that we can get a lot of structural information on the glycopeptide, because at least from my experience, the slight structural difference on the glycopeptide usually doesn't give you enough leverage in ion mobility to actually be really different. That said, there is a possibility to do this on the fragment level, and that's something that we and others published earlier, where you can actually look at oxonium fragments, for example, to differentiate to 3 and to 6 sialic acid on glycopeptides. So you can actually get limited structural information, or at least of epitope, structure epitope information in a glycopeptide-specific manner, if the right tools are there. But let's be fair. 20 years ago, a little bit more than 20 years ago, when I started in that on there, the tools that we have now available looks like for me like something from Star Trek in 300 years compared to what we had 20 years ago. So who knows what in the next 10, 15 years will be available to us that we don't even dream of at the moment. So.
You mentioned the rearrangement of the glycan can happen. So can you comment what precaution we should take to avoid this kind of confusion?
Acetylation
Yep.
can essentially, can definitely do that. But again, you don't necessarily want to do this on a complex sample because you're gonna probably introduce more artifacts than you actually want to. I think it's more essentially putting up the attention that just because you have a fragment doesn't necessarily mean it's this particular epitope. So just because you have a 512 fragment in your CID spectrum doesn't mean you have a Lewis epitope on your glycopeptide if you just have a core fucose there in the first place. So that's essentially, it's more raising awareness when doing the data analysis and not just relying on certain fragment ions that might indicate something, but that might also not be completely specific for this particular purpose.
Yeah, thanks for the great talk, Daniel. So, I mean, we use a lot of mass spec, and mass spec is not that good in differentiating isomers. So I was wondering, how much of the human glycome do we grasp and cover with the methodology we have, and how often are we misled in terms of isomer assignments?
I think in terms of isomers, thanks Manfred, we are still missing a lot. And they're not all technologies give you the resolution for different isomers in the way that you want it to, especially not mass spec, especially not if there is a huge mixture of them there. So that's at least my experience, and that's where orthogonal method come into place, like combining LC, like porous graphite carbon, which I think you're gonna mention a little bit, on there, which is the method of choice in my lab for most applications. But yeah, that doesn't mean that's the only one we are doing. I think it's also raising the awareness that there are isomers out there, and that goes back to the data analysis and to the data reporting point of view. Just because you have a certain composition, and just because the 2,6-silated N-glycan is the one that is most reported, doesn't mean that it's actually in your case, and it's most likely not in your case if you're looking at certain tissues or looking at certain aspects on there. But that's where the reporting again comes into place. And I think it's also our responsibility here to, as glycobiologists in particular, to report this as accurately as possible and not overinterpret it, just because it would nicely fit with the theory that we are having, unless you have orthogonal confirmation by different methodology to give you that information.
I would like to add 2 points. I think I agree with you. I think with current modern large-scale glycoproteomics, everything is biased. Especially, you found that they report that the majority are high mannose.
Yeah.
Okay, because essentially the high mannose peptides are easier to
fragment,
enrich, easier to fragment, easier to identify, easier to pass the threshold, etc.
And you just have essentially talking about 5 or 6 different masses that you have to consider, not the other 500.
And I've yet to see those large-scale proteomics, glycoproteomics, that report a very complicated structure on the N-glycopeptide, let alone O-glycopeptide. Okay. Yeah. So the second point I want to point out is that we do have a glycoproteomic workshop on Wednesday afternoon. Please join us. And Daniel mentioned the first HGI initiative, and that's when Stacy is going to introduce to you the second initiative. And we also asked some developers of software to join us. You can challenge them if you want. But to me, is that all the software work to a certain extent and doesn't work to a certain extent. And don't believe what you see. And I think we still need to go back to manual examination.
And maybe one more thing also, it's not to bash the software developers, but the software is just as good as the data is that you produce in the first place. So if you do not take enough care in producing good data, software is not magically making bad data great.
Particularly the glycopeptide spectrum. If you don't have any BYI, and how are you going to ID anything confidently? Okay. So I think with that note, we're almost on time, and we have a break, and then please come back at 11:10. Yeah. To the last talk of this session, and in fact the last talk of the whole entire series of our advanced courses. And it's our great pleasure to have Manfred here to give us the last lecture and the final word. Yeah, and I know Manfred for a long time actually, dated back from the time that he works with Rudolf Geyer in Gießen in Germany.
Yes, indeed.
And I think I met him in one of those international glycoconjugate symposium. I can't remember which one.
We also in Florence together, yes.
Yes. But then after he got his PhD, then he moved to Leiden. And he has stayed there since, right?
Yes.
Yeah. But today he is the professor of analytics for biomolecular interaction at the University of Amsterdam. And he is also the head of the Center for Proteomics and Metabolomics at the Leiden University Medical Center. So it's a great pleasure to have you to tell us what you want to say. Thank you.
Thanks, Ku, for having me here. I'm excited to be in Taiwan to explore the surrounding here whenever possible with the intense program here. You're an amazing organizer, I have to say, and that makes me looking forward also to the rest of the week. It's all perfectly organized until now. So my compliments on that. And it's an honor for me to be here and presenting this in this session on glycomics and glycoproteomics, also together with Endel. And I have to say the work by Anne and by Ku was inspiring when I started as a young PhD student. And in that respect, it's a very great honor to be here with you guys. Okay, so when we planned this session on glycomics and glycoproteomics, we agreed that I would look more into applications and 3 specific angles, and those are mentioned here. So on the one hand, it's the intact proteins. On the other hand, higher throughput, so analyzing hundreds or thousands of samples, and then also looking in tissues at spatial distribution of glycosylation features. I will start with the spatial distribution, so the mass spec imaging of glycans. Because that's what you want to know. We heard about the different cell types, different tissues. They are different in terms of glycosylation. So you need that resolution. You have to know what you are looking at. Is it a neutrophil? Is it a macrophage? Is it an immune cell? Is it a fibroblast? Ideally at a single cell level. And this slide just shows for a cancer, for colon cancer tissue, a tumor. So it shows that if we take a specific, I'm struggling a bit here. If we take a specific glycan, we see its spatial distribution. So you do a mass spec per pixel. So you have here thousands of mass specs for each of the tissue regions, kind of at a roughly single cell level. And then you see the high mannose glycans. You see here 3 different complex type glycans, diantenaries, and here you have 2 times the 2,3-linked sialic acid, which looks to the left, and 2 times the 2,6-linked sialic acid looking to the right. We do some chemistry to differentiate these isomers, and what you see is their spatial distribution is very different for this one and that one. And then if you overlay them in red and green, it becomes apparent that indeed very different regions of the tumor express the one or the other glycan. So we take this as a justification also to do some chemistry. In this case, we are lucky. So we have the 2,3, 2,6 sialic acids, and there's chemistry, which also came from the lab of David Harvey many years ago. And we use this chemistry now on the tissue to differentiate these glycan isomers. And really, by that, if we link up with the pathologies, we can tell that the red signal, that's kind of fibroblasts, stroma, and the cancer cells, those have the green color, so they have the 2,3-linked sialic acid. And this is just one example why it matters to differentiate isomers, but also to look specifically at one region of a tissue. Now, if we look at the entire workflow, how would you do this? So you would have a tissue block, which is formalin fixed and paraffin embedded. That's what the pathologist will do normally in a clinical setting. You then cut it and mount it on a glass slide, and then we do some chemistry on this glass slide. As I said, for the 2,3-linked sialic acid, we do one chemistry. For the 2,6-linked, another chemistry. And it gives different masses, so we don't have isomers anymore. We have now distinct chemical entities. And then we release the glycans. But what I want to stress here are two facts, and those are highlighted down here. So one is solid phase chemistry. So we have the glycans still on the protein, which is fixed by this formalin treatment. So the protein sits there, and we can do this very nice chemistry to differentiate the sialic acids. So that means we don't lose any spatial resolution. Once you come in with the enzyme and you cut off the glycan, you have lateral diffusion, so you can lose spatial resolution, which you don't want because you want to have this single cell resolution. So this is a perfect match, let's say, that we can do all this chemistry while the glycan is still sitting on the protein. And the other one is indeed, these tissues, these are treated by the pathologist to do H&E staining, their typical historical staining patterns. But this is a perfect fit with our glycan chemistry because what they do is they wash out lipids, they wash out metabolites, they cross-link proteins. So virtually all the analytes are almost useless, let's say, or gone. But the glycans are still sitting there, are not affected by the treatment, so they are perfect analytes to analyze on these pathology sections. Good. So just one perspective where glycomics can come in. They are stable. You can use libraries which are 10, 20, 30 years old of pathology tissue sections. So all the pathologists have these in their cupboard, and you can still probe the glycomic distribution in those historic tissues. Now, if you do that, you will try to relate this to what the pathologist has found. So here, this is what you normally would get. So the pathologist will tell you, here you have one region, another region, and still another region of the tissue. So for me, it all looks kind of the same. But those guys, they have, of course, the training to tell you, this is what you're looking at. And in the end, it's, of course, about... why is it not moving? Maybe. Okay. So it's about then having our mass spec imaging glycan data. So here you look at the glycans, and we can look at two different glycans for the glycan isomers, as I showed here. We can look at glycan traits, so kind of ask what is the level of cofucosylation, ternary fucosylation, how many diantennary, triantennary glycans do we have. So you name it. You look in different ways at your glycomic data. You can do a multivariate data analysis in a spatially resolved manner. So then you ask, where do we have certain multivariate features in the tissue? And then in the end, and that's here at the right-hand side, just in the legend, in the end, you want to link the histological assignment by the pathologist with your glycomic assignment. And finally, you hope that you have added value, that you don't only merely see what the pathologist anyhow resolves, but maybe something which looks by conventional H&E staining homogeneous will now look like a gradient, and you will see a molecular gradient, so new histological features encoded by glycans. And just to make things a bit more complicated, what else can you do now with this old-fashioned moldy glycan analysis if you do it in a spatial manner? What you can do is you can also, if you still have a tissue which is alive, you have maybe a kidney, a live kidney which you section and which you mount on a tissue, you can do some metabolic feeding, and that's indicated down here. So you have a mouse, you have a kidney, you mount the kidney on the tissue, the carrier, and then you come in with labeled compounds, and then you can do some
metabolic tracing. So you can see C13 glucose, where does it go, into which metabolites, where does it go, into which end glycans. So we kind of can have a temporarily resolved glycome picture. And this is still future plans, but you can do this for lipids, you can do it for metabolites, and you can do it in a multi-omics manner, we think, for looking into metabolite labeling, but then also glycan labeling. So this is the lucky situation. For end glycans, we have the enzyme. We can do all the sample preparation on the tissue, and we can use a simple multi-mass spectrometry to probe the spatial distribution. Now for O glycans, we discussed it already. We need, Henrik also mentioned it, and Daniel, we need enzymes. The enzymes have selectivity. The enzymes can hitherto not be used with mass spec imaging workflows. So there we would use another approach, and that's shown here. So we would still want to look in a specific manner for a specific cell type in the tissue, what is the O-glycan there for those samples? And we want to differentiate isomers. And I will just show one example on that. So again, we start with a histology. So we have a tissue which is mounted, but now we can't do the mass spec imaging approach. What we would do is use laser capture microdissection. So you use a laser to cut out a specific region of the tissue, and that's shown here with the green borders. So you cut out this epithelial layer of a colon cancer, for example. So this is the healthy epithelium, and down here you cut out the cancerous part, and then you can compare healthy versus cancerous epithelium cells. And then a workflow. This is now a PGC workflow, which Daniel already presented, so the PGC method. So what we do in this case, we cut out the tissue, we prepare it on PVDF membranes, so you immobilize the glycans, you cut off the end glycans, you then do a chemical release of the O glycans, and you have glycan alditols in the end, and analyze them by LCMS. And I will show some data on this in the next couple of slides. First, on method validation. I think that's an important aspect. So maybe we have indeed those massive biases which you mentioned, Anne, for native glycan analysis. In this case, it's not MALDI, but electrospray ionization. But what I think is even more important is that it's repeatable. At least what we get is consistent, and we get it again and again. And that's shown here. So using this workflow, we can separate end glycans, we measure them by mass spectrometry, and we can get the profiles, and we get them in a consistent manner over days and weeks. And we also do the very same for O glycans. Also there we have repeatable profiles. And then we can start compare different tissues. And the nice thing is here is we can now, for the very same patient, for the very same section, take the healthy epithelium, we can take the cancerous cells, and that's shown here, so cancer versus normal cells. And then you see some differences, and those are highlighted here. Maybe you can see that. So we have some glycans which we only find for the cancer cell, but not for the healthy colon. So colon cancer, the cell changes its behavior, its gene expression, all of that, and it changes its O-glycone. And if you look at these glycan structures, you see it's core 2 glycans, and they have a bunch of Lewis motifs here on the 6 arm of the Saleh-Lewis, but also Lewis epitopes on the 6 arm of the core 2. And of course, you don't want to do this only for one sample or one pair of samples. You want to do it for a bunch of those. So Anne mentioned analyzing 40 samples, for example. Here we only did maybe 15. So we analyzed those samples, we compared them, and what we saw is the normal mucosa and the cancer are very different. Red is different from blue. So really, you have the differences between individuals, but the difference between cancer and the control tissue is dominating here these profiles. And there's different ways of looking at these data, and that's also an important aspect. You have to choose for ways of analyzing the data. So here we just looked what are now the expressed O glycans for the different tissue regions. And so for the cancer, that is these first bars, we have 100 glycans we only observe for the cancer. They don't show up for the control tissue, also not for the stroma, for the fibroblasts. So those are cancer specific in our assay. It's 100 of them. And then you can see how do they look like, and we see it's core2 glycans, and they have a lot of 2,3 citylation. And we explored this further. But I have to say we were also really extremely excited about those data, because I started with this expecting that we might have grayscale differences. So same glycan, maybe with upregulated, downregulated for cancer versus control. But the differences, as I said, were black and white, 100 glycans only for the cancer. And this is then a way we decided to visualize these data. And that is, this dot indicates this glycan is not in the healthy colon. It is present in the cancer, only in the cancer, not in the healthy colon. And then if you stratify for the aggressiveness of the cancer, then it's mainly in the aggressive forms and less so in the non-aggressive forms. So really a marker, a O-glycan marker of aggressive colorectal cancer. Just a different way of visualizing the very same data again. So it's these core2 O-glycans with the silyl Lewis motifs, which are present in the cancer but not found in the healthy colon. And then this is, of course, a very relevant way of looking at it. So if we speculate about biosynthetic pathways, how do these O-glycans occur? So we start with the O-GalNAc, we have the core2 buildup, and then on the 6th arm we get the silyl Lewis motifs. We also looked at transcriptomic signatures, and those very much align. So indeed, this is the transcriptomic program which you observe in colorectal cancer, but not in the healthy epithelium. So, and actually we started with this simply with a biological question, and we thought maybe there's some diagnostic potential, but now we think with this rather black and white signature, hopefully these structures up here can be targeted with therapeutic antibodies, because we expect them to be at the cell surface, and hopefully this is a new therapeutic avenue for colorectal cancer, hopefully also for other epithelial cancers. So this was the spatial aspect of my presentation. Now the second part I want to talk about is intact protein monitoring, and also Daniel mentioned that. So Daniel's talk was very much focused on bottom-up. I think it's very powerful. You get these thousands, 10,000s of glycopeptides, you can assign them. The software is obviously the big bottleneck we're tackling there. But I think it's also very powerful to keep your molecule intact and hopefully get a comprehensive overview on what is on my protein and how is it affecting properties, interactions of this protein. How do the glycans come in there? And that's now, as I said, the second part. I will stick to antibodies, also shortly mentioned by Daniel. If we look at the antibody, we know that in the CH2 domain, we have this conserved N-glycan. This N-glycan can be very diverse in structure, and then we have different motifs on the N-glycan, like the co-fucose, the galactoses, the sialic acid, and we have quite some knowledge on how these tiny, small glycosylation changes affect the properties of antibodies and the effector functions. And some of that is summarized here. So what we did years ago, together with Gestor Widerson, our collaborator at the blood bank, he made 20 different glycovariants of an antibody. So with a fucosan, lacking the fucose, with different levels of galactosylation, different levels of bisection, silylation, so all the variants you could imagine, and he tested them in a simple binding assay. So how good do these antibodies bind to the 3A receptor? So this is kind of an immunological junction. You have the NK cell where the 3A receptor is there, and it interacts with the Fc portion of the antibody. And for example, this is then used to kill, to internalize a virus, but also to kill a cancer cell. So the cancer cell is attacked by an NK cell, and the antibody makes the functional bridge. And then what we see here is when the co-fucose is present, actually this will not happen because the antibody will not engage with a 3A receptor. Whilst when the fucose is lacking, you have a very potent antibody engaging with a 3A receptor. And using in vitro assays, we also showed, in this case, we have cytotoxicity, so the NK cell will kill the cancer cell once you give the antibody lacking the co-fucose. And of course, having the relevant binding to the cancer cell. Now, a way of doing intact analysis, intact glycomics or intact protein analysis, I should say, is now is something I want to present here, and it comes with an overview of all the glycosylation features we have, or most of the glycosylation features of the antibody, but also you can do a functional probing of these glycoforms. So what we did here, in a nutshell, is we used a recombinant antibody with all its variations, all the different glycoforms, and we immobilized the 3A receptor on the column, on an HPLC column, and we flush now the antibody across this receptor, and we see that some glycoforms elute early, are hardly binding, and these are the ones with the co-fucose present. Whilst indeed we kind of recapitulate what we did previously with the 20 individual glyco-engineered forms. So now we see indeed those glycoforms lacking a fucose on one or the other branch are the ones engaging with the 3A receptor, eluting late, reflecting high affinity to the receptor. And then you can look at fine structures. You know, here you have two different peaks, so probably we are resolving some glycoisomers here at the intact protein level. And the setup just uses the mass spec of this intact protein to probe the glycoforms, to resolve all these different glycoforms, and indeed also indicate their relevance for the binding to the 3A receptor. Okay, so just this is a kind of a background. So glycosylation of antibodies, we know that, and many people have worked on that, matters for the biological activity of an antibody. And we looked into rheumatoid arthritis as one of the disease models where we know antibodies are important because antibodies are there for in the synovial fluid. So there you have the have also B cells. So you have locally in the joint B cells producing antibodies, and these antibodies are specific. So you have them in and sensitive. So you have them in many of the RA patients. 70% of the RA patients have these autoantibodies against a modification of the protein. So many proteins are modified. Arginines are turned into citrulline, as shown here. So that's the motif. You have it on many proteins, and there you have this autoantibody production, and these autoantibodies are drivers of the disease. And then we were wondering, do they all look the same? Do they look different? What is their glycosylation? What are the properties, molecular properties of these autoantibodies? And for that, I want to present now a workflow, kind of a middle up, so almost intact. So we decided to get rid of the Fab portion of these antibodies and focus on the Fc portion. So just the part of the antibody which engages with the receptor. And we did this for 11 patients. We looked at the site of inflammation where you have the autoantibody production, but also systemically in serum or plasma, and characterize these autoantibodies. So this is in more detail the workflow. You cut the antibody in the hinge region, and then actually you first capture the antibody, and then you cut it, and you do that for the specific autoantibodies, but also for total IgG. And then you have these Fc half portions with the glycan on, which you then measure by mass spectrometry. And the nice thing here is you get glycosylation information, but you get way more. And I think that's important. Many molecules we look at are glycosylated, but they also have other properties. And it's important to see glycosylation in the context of other omics. And here we look at glycosylation in the context of IgG subclasses. So is each subclass equally represented? And that's what we see here. It's not. It's IgG1, which is dominant for the autoantibody, and not the other subclasses. Because we have mass spec, and the mass spec tells us this is the mass of this Fc half, and it tells us this is IgG1 versus IgG2, 3, and 4. So IgG1 is dominating these autoantibodies. For another patient, we see heterogeneity. So we have two gene copies. We are heterozygous, or at least this patient is. So two different allotypes of the IgG1. And here we see both allotypes are represented in these autoantibodies, but the ratio of these light blue peaks is different for the autoantibody compared to total IgG. So two gene copies and one gene copy contributes more to the autoantibody than the other. And you can quantify this for different patients, and consistently we see that the ratio of these allotypes is different for the autoantibody compared to total IgG. It's getting complex, I have to admit. But the basic message is you can resolve heterogeneity of the protein portion, subclasses and allotypes, how they contribute to different disease processes. All of that is resolved. And then on top, you get the glycosylation information. So it allows you to get glycosylation information knowing all the other properties or many other properties of your molecule. And here we just, bless you, here we just look at the glycosylation heterogeneity, and what we see is total IgG in blue is different from specific autoantibody glycosylation. So total IgG and autoantibody are different, and then with this multivariate data analysis, you can look at the loading plot, you can look which glycosylation features are contributing, are dictating now the glycosignature of these autoantibodies. And I think it's easier to look at that in these plots. So what we see is, for example, that fucosylation is different. So antibodies are highly fucosylated, but what we see is that these autoantibodies are virtually completely fucosylated, whilst a lot of other antibodies are diverse in terms of fucosylation. And we can look at bisection, we can look at site occupancy. So we now resolve differences for these autoantibodies versus total IgG. And we can do that for total, for the circulation, but also for plasma. We can compare plasma and synovial fluid glycosylation features, and here we mainly see that galactosylation is different. So the autoantibodies at the site of production, at the site of inflammation, have a lower galactosylation as compared to the ones in the circulation. We can look at other sites of glycosylation, so we have also on IgG3 a second site of N-glycosylation, and just to show you that we can now measure this extra site of glycosylation on top, so we get CH2 domain, CH3 domain glycosylation, and also there we see that the autoantibodies are different, and this is the same for plasma and synovial fluid. So I have to admit it's a vast amount of data, but the basic concept I want to convey is if you can, don't look only at glycosylation, but look more broadly. So what is my molecule looking at? How is it interacting? Maybe this is dictated by glycosylation, but maybe it's the allotype, or it's a oxidation, or whatever modification of the antibody. So I think this comprehensive glycoproteomic analysis of a molecule is very, very insightful, and can avoid us making false conclusions. So it could be glycosylation, it could be anything else, it could be the interaction. So if possible, probe these modifications together. So that was the second part of my presentation, indeed, on the intact analysis. So we started with the spatial resolution, we moved to the intact analysis, and now it's about numbers. So analyzing many, many molecules. And before I dig deeper into this, I have to say it comes with compromises. So if you increase the numbers, you lose depth. So you can't do it all. You cannot separate and isolate and characterize all isomers. So it's painful to do this higher throughput analysis because you have to stop doing other things. You can't separate all isomers. You can't look at the tiniest peak because otherwise you never get done. So this is something indeed which Anne mentioned with the 40 samples, because you were discussing then all the confounders. Yeah, are the women older, younger, which blood group? At which gestational moment are you looking at it? So you easily come up with 10 variables, but you have only 40 women, and you have maybe 100 glycans. So it's getting crazily complex, and you enter the danger of overfitting, and that pushes you towards analyzing hundreds or even thousands of samples. So you have the power to still conclusively find out it's this feature. I'm pretty sure it's this feature which dictates my separation and not the other one. So that's why you would go to higher throughput. And for that, I want to start with COVID. So when the pandemic hit, we started wondering, what do the antibodies look like? Because it's, for a big part, immunity against COVID is about antibodies, IgG1 antibodies. They bind to the receptor binding domain or the S protein on the virus. I don't have to tell you about that, I guess. And then we were just wondering, what is the FC modification of these antibodies? So what we did is we had plasma from patients which were at our hospital in Leiden. We used a microtiter plate adsorption assay, meaning the S protein is coated on the ELISA plate. The antibody is bound from serum or plasma, we wash, and then we don't do an ELISA, but we elute the antibody. We digest it, so we generate a 9 amino acid FC peptide, so the peptide portion of the FC domain with a glycan on, and then we measure that by mass spec, in our case LCMS. And what we saw is just for one patient shown here, so the total IgG is not really is not that spectacular, and it's also constant over weeks. So this patient came in with a serious COVID-19 infection, otherwise you wouldn't get into an academic hospital. And then after some weeks, the IgG profile, glyco profile, looks the same. Now, if we look at the anti-S profile that's shown here, now what we see is that some glycoforms like this one here lack the core-fucose. So you right away, if you look at all these glycoforms, you see, do you see the cursor here? Not really. What happened? The computer is thinking. Anyhow, you see some glycoforms lacking the core-fucose. So this means, indeed, I told you core-fucose is important for dictating NK cell activation, macrophage activation, inflammation. So we think these glycoprofiles could possibly explain inflammatory responses. So an inflammatory signature when the patient comes in, and after some weeks, this is resolved. We then had the chance of following these patients in very great detail. So what we saw is that fucosylation, when the patients come in, was very low, and then within days, we saw that the fucosylation was normalizing, was going up. And we were shocked by this data because we know antibodies have a half-life of 3 weeks. So what we expected is very slow changes, you know. But I think what happens is these patients come in, and the very first antibodies produced have this low fucosylation profile, but then after a few days, other B cells come in, and those new B cells produce new antibodies which have high levels of fucosylation. So a very, very quick change of the glyco setup of these antibodies. So this was against textbook knowledge, I would say, but you can explain it. So indeed, very dynamic changes of antibody glycosylation. And then from there, we moved to in vitro assays, and we asked, are we back? So this is what I told you. So very fast glycosylation changes, you know? Look at this. You follow patients, and within a few days, you see already that the glycosylation is moving in certain directions, way faster than you would expect. And we saw that for fucosylation, galactosylation, silylation, and bisection. And actually, before I move into the functionality, what I want to say is we see this for more diseases. So we had seen this for other diseases, for other immune diseases, for various human immune responses. We see these low fucosylation responses. But indeed, we also saw it for COVID. And we even see it for transplantation settings. So also there, it's probably very interesting to look into antibodies causing complications with transplantation. So this fucosylation kicks in at various levels. But now back to COVID. So we have this low fucose response against the surface of the virus. And then indeed, as expected, we showed that these low fucose antibodies are potent in inducing inflammation. It's a simple in vitro assay, and you look at interleukin 6 production. So these are the ones which could maybe in the lung cause inflammation and drive pathology of COVID. But then if we looked at associations with the outcome, so do patients have to get intensive care? That's the red ones, or not? We saw that it's actually the galactosylation, silylation, and bisection, which at the moment of hospitalization tell you this patient will need ICU care, yes or no. It's not the fucosylation, and we're puzzled by these results. But also, I mean, this is what we did on infection. Then we also looked into vaccination, but in view of the time, I think I will move on. Just what I want to tell you is, I think we have now these assays to analyze hundreds or thousands of samples. We can do that for various infectious diseases, various autoimmune diseases, and so on. And we can assess this fucosylation, and we see that in certain contexts we get this low fucose response, in other cases we don't get it, and we think we have kind of a double-edged sword, you know? So we think in some cases it's very potent to have this low fucose response to, for example, kill a cancer cell. In other cases, it causes massive inflammation and maybe lung damage. And we think it's important to get this information, but ideally we could also intervene and change fucosylation levels at a certain moment. Now, in the very last part, I want to show analysis of, again, hundreds or thousands of samples, just a variation of what we just did. We look at blood in this case. So what we are analyzing is just serum or plasma, all the proteins in there, that's antibodies, but that's also acute phase proteins of the liver, and we look at their glycosylation. And we have a robotic assay there, which allows us to analyze hundreds of samples per day. And we again use this salic acid differentiation. So we now can see 2,3 to 6 salic acid, and we can look at those specific changes. And this is MALDI mass spectrometry, and we can do this in a very repeatable manner. N. Maybe not accurate values, but at least it's repeatable. And we did this for liver diseases, so fatty liver. This is a huge problem in the world. We all eat too much, we get fat, and our liver gets fat. And what happens then is you have this liver fattening, and in the end, you have liver fibrosis. And that's the different fibrosis stages. You get liver cirrhosis, so the liver is not functioning anymore. You get hepatocellular carcinoma. So this is really a cascade of pathologies. And we did two cohorts, a discovery and a replication cohort on this. But basically what we found is it's all about silylation. So when the liver doesn't function properly anymore, IgG silylation is total silylation is changing from the one to the other phenotype, and that's shown here. So we have an increase into 6 silylation with liver pathology. And this is virtually the only change we saw. All the rest is the same, but just the salic acids are flipping from 2,3 to 6. And we saw that in the discovery and in the replication cohort. And the astonishing thing is, if you just look at this 2,3 silylation, it allows you, on tetrahydrogen glycans, it allows you to almost black and white differentiate between people with fibrosis and people without fibrosis. Until now, you have to punch a needle into the liver, and you get out a tissue section, and you do histology. But we think with this simple assay, you can also really pick up liver fibrosis. Now, of course, you can do this for liver diseases, but you can do your blood glycome, serum plasma glycome, for a bunch of diseases. So just, I'm wrapping up, just to show for cancer, for inflammatory bowel disease, for rheumatoid arthritis, for a number of diseases, we have all of these glycosylation changes. And you see some arrows going up, other down. So it's not like that, like each disease would have the very same phenotype. You can then do heat maps like shown here. You can link it to specific processes or molecules. You can compare aging and high BMI with disease signatures, so you can try to understand what is driving what. And you have to do this on hundreds and thousands of samples, otherwise you don't know, you can't do all these corrections and you don't know what you're looking at. And we did this now for a bunch of different diseases, and I think this is another bioinformatic challenge, how to link all these data. So you have all these different disease dimensions, and we try to integrate all these cohort studies to find out with inflammation, with different types of cancer, with different types of infection, do we have very distinct glycomic changes? And of course, eventually, we want to link this to the carrier proteins and understand also the regulation, the mechanism which is underlying. Now, what is nice about this is you can do this from serum or plasma, but you can also simply take a dried blood spot and you get the very same profile. So from one blood spot on a filter paper, you get the same glycome. So that means you don't have to go, you can also sample at home via finger prick. And it's robust, so you can store dried blood spots for weeks at room temperature. Glycans are stable. You always get the very same glycome. So this comes with clinical implementation, clinical optionalities. So just to summarize, so one dimension is cell type specificity or tissue specificity, so you have to know what you're looking at. And for that, mass spec imaging is very useful. Sometimes you have to take extra efforts like laser capture microdissection, but it's important that we know what is the cell type we're looking at. Otherwise, we are just blurring the whole glycomic picture. The second aspect I came up with is glycomics in the context of proteomics, so intact protein analysis, also probing your protein in a functional assay, and ideally you get the structure and the functional analysis together, and indeed intact protein analysis is there a very powerful approach. And now the last part I was showing was higher throughput. It's always painful because you lack depth, that's what I told you, but you get coverage of different diseases. You start resolving how the glycome is changing with age, with sex, with hormones, with different diseases. And I think that's very valuable that we get at least this map of the human glycome, how it might be regulated. I use this slide here kind of as an anchor, and I want to give two perspective views. So I think mass spec is a very powerful method, certainly if we combine it with mass spec imaging, spatial resolution, also ion mobility, some spectroscopy, and I think the key challenge is indeed bioinformatics. So how do we process these data? How do we assess glycoproteomics data? So we really have to focus on that. And then in terms of functions, I think it's very important to integrate different omics, not to look at glycans only. Microarrays are powerful. We can now do molecular biology, so we can intervene. We can change glycogenes, expression. And there's a bunch of very promising chemical biology tools. And I hope that all of this is being integrated and will bring us further. And with this, I want to conclude. I want to thank my team, my collaborators, and I want to thank you for your attention.
Okay, thank you. Any question for Manfred? Any food for thoughts before we have lunch?
Okay.
Yeah, I have a question regarding the imaging. So do you think, or what do you think is the best way to do like the structural assignment of the N-glycans? Because like especially of the lower signal to noise
yeah, good point.
signals, yeah.
So one challenge is the lower signal noise. So there, if you push the spatial resolution, you lose your depth in terms of mass spec imaging. So you have to ask yourself, do I need single cell resolution? So if you don't need that, just sample maybe a bigger area of the tissue, kind of 30 times 30, 40 times 40 micrometers. By that you get more signal and more depth. So that's one approach, of course. And then one challenge is the isomers, because mass spec imaging will not resolve isomers. I mean, we have the salicylic acid linkage derivatization that helps, but there's many more isomers. And I think the ion mobility, which you have on the Bruker system now, is very promising. So it will tell you at least that you have some different isomers. It will not resolve per se all their structures. And then what we think for mass spec imaging, you should do all the work on the tissue, but you have to take your glycans off. You have to use your best methods, like in our case, PGC, to also do an in-depth analysis and link the detailed data which you have off tissue with the data you have on tissue.
Yeah, like prepare a library and then use that.
Yes.
Yeah.
So you define your library off tissue and use that as a framework to do your mass spec imaging analysis. So integration of different approaches, that's key, I guess.
Thanks.
Okay, based on my understanding of your talk, so one protein can have a different glycan pattern, right? So how exactly this glycan pattern is regulated?
That's a very good question, I think, yeah. I think we have to stay... Yeah, but I mean, we have to understand that. Also, if you want to come with drugs, if you want to change it, you have to have an understanding of the regulation. And I think we know there's glycosyl-transferases, but then there's many different things going on. There's metabolic regulation. There's many layers we don't understand yet. And so that should be, I mean, the one thing is, what do glycans do in terms of changing the properties of the protein, but indeed, how is it regulated? It's a big area of research, and we're just starting there, I think. So like for these liver glycomic signatures, they are all there, but how are they regulated? We don't know.
Okay.
Thank you for the nice talk. So my question about the MS imaging. So that after the PNGS treatment, the glycan were observed directly with no modification at the non-reducing end, right?
That's what I showed now. Yeah, indeed, without reducing end modification. Yeah.
If you put some modification at the non-reducing end, the higher intensity is higher.
Yeah, I mean,
is it difficult?
Yeah, I think that's something to consider, to do a modification, kind of a reducing end tagging of glycans. But the problem with the mass spec imaging is every step you include on top might be incomplete, so you generate heterogeneity, and it may compromise your spatial resolution, because you have to get your glycans into solution, and by that they might diffuse. You want to comment on this, Daniel?
Yeah, and also it's more important for the imaging to actually see what are the differences within the tissues, not necessarily to differentiate all the structures. That's what you would do offline. And if you really want to notice and understand this from that point of view, I think for the imaging, the power is really understanding the spatial difference between structures.
Thanks.
In your method, before the PNGS treatment, you add the...
we do chemistry on the salicylic acids, yeah?
I see. Thank you.
Yeah, that's indeed what we are doing. Yeah, and I think that's indeed very powerful. It's a perfect match with this mass spec imaging workflow because the glycans are still immobilized and you keep your spatial resolution. There's one more question here.
Yes, I have a question regarding the time. Do you know when these alterations occur? Is it like early during disease?
Which alterations?
The glycosylation, like when you compare healthy tissue to disease like cancer tissue or something. Yeah. Like, does it serve as a biomarker or is it like late when the disease is already like progressed?
I mean, often you don't get these early samples. Let's start with that. So that makes it hard to address that. Sometimes you have these samples, and sometimes the data indicate that glycomic changes occur already before the disease. So I think there's no easy answer to that, but it's not like they would always just follow the disease. And I mean, we now have a lot of data also showing that glycans are driving disease. So I think, indeed, they are just, in a way, they are often an integral part of the disease process. I think that's maybe the best way of looking at it. So often they can serve as very early disease markers.
Yeah, thanks. Very inspiring talk. I have a question regarding the MS imaging. So if my interest is the terminal glycan epitope like Louis X or Star Louis A, can MS imaging tell me the distribution abundance of those glycotope regardless of the antenna or backbone structures?
Good question. I think I would often, whenever it's about glycan mass spec imaging, I would always think, aren't there easier methods which give me the data? So maybe you can use antibodies and immunohistochemistry. So it's very tedious. It's a lot of work. It's a lot of data. So I would only do glycan mass spec imaging if there's no alternative approach, which gives you the answers you're looking for. Okay, that's all.
Yes, I have a question about the mass spectrometry imaging. Because you use like a PNGS for treatment, is it applicable to O-glycan, like you do treatment with enzyme specific for O-glycan and do analyzation?
So for the O-glycans, we discussed that in part. There are some, you could generate some glycopeptides. You have some release enzymes, but they all come with a massive bias. And there is no good workflows until now for O-glycan mass spec imaging. So that's why we would also go for this laser capture microdissection approach, yeah. So it's only, until now it's only N-glycans, it's glycosphingolipids where you can do mass spec imaging. The other things are emerging, but not there yet for routine use.
Thank you.
Okay, I think, thank you, Manfred. So before I pass on to Anne to say a few last words, I think we have a fruitful discussion. Obviously, we cannot cover all aspects of the glycan analysis, particularly glycomics analysis. Glycopeptide is a little bit simpler because everybody cut their protein into glycopeptide. Okay. But glycan, you have derivatization, you have reducing and reducitization, and you have the non-derivative, and you have PGC, you have all these two-dimensional mapping, etc. So one last thing I do want to say is that we all stand on the shoulders of many giants in the field. And one I can think of, which I want to mention, is that when we talk about permetylation, you should know that Hakamori invented permetylation. And he is the one that I would like to dedicate it to him. And through the metallation, we saw Ben Lindbergh, etc., they invented the linkage analysis, which people don't use it anymore today, mostly. So I think it's good that people can dig into the literature, particularly the younger generation, to know where we are now and to appreciate our past. So with that, pass on to Anne to say a few last words.
Thank you very much, Ku. I just want to sort of reiterate and emphasize the need to not just use moldy electrospray, etc. GCMS can be extremely valuable, and instruments are really, really sensitive these days. So linkage analysis we often don't use very much for mammalian glycans because the biosynthetic pathways are so well worked out. If you're prokaryotic or archaea people, you almost certainly want to invest in a GCMS as well, just to highlight that. The other little thing I'd like to say before I thank my colleagues and all of you as well is I was, where's Manfred gone to? Oh, you're over there. It was really nice that you talked about rheumatoid arthritis because, again, standing on the shoulders of giants, the other really key person in the early development of the glycan analysis techniques that I would like, I mean, there's so many people I'd like to mention, but I would particularly like to mention Akira Kabata, who was the lead person in Japan for so many years and still active. And it was the work on rheumatoid arthritis that he did in the late 70s, early 80s on IgG, and in collaboration with the Oxford Raymond Dweck and Tom Rademacher groups that led to the development of many of the methods that complement the kinds of things I talked about, but in particular actually led to the word glycobiology. So Glycobiology Institute, Raymond Dweck established in Oxford, that came particularly with funding from a company some of you might have heard of, it's not called it anymore, Oxford Glycoscience, to exploit what had been learnt about rheumatoid arthritis. So it is, in terms of history, please, if you're younger people in the audience, have a look at the early papers, because many of the questions you're asking today, particularly about imaging is kind of where glycobiology in terms of analytical glycobiology was in the early to mid-1980s. So a lot of the things that you're asking, you need to be thinking, why am I asking these questions? In other words, why am I applying the techniques? There are so many different techniques that can be applied. You need to know what is the answer you want to get in your experiments, and that's the key message that I'd like to give to all of you. That because you can seek information so much more easily today than you could 20, 30 years ago. You can send emails, you can do social media, etc. But you need to know why you want the analytical data. That dictates which experiments you do. So what I'd just like to do is to thank Daniel and Manfred for being such wonderful colleagues for this morning, and I think we integrated our talks rather well, especially when we hadn't shared anything in advance other than a quick Teams meeting on what we were going to talk about. And thank you, the audience. So many of you asked such really nice and absolutely excellent questions that kind of hit the nail on the head for many of the techniques that we use. And I've loved the morning. I've really learned a lot, actually, from the other talks. And I hope that I will have a chance to chat to some of you between now and Wednesday morning. I actually am going back to England on Wednesday. So if you want to catch me before Wednesday, but I will be actively participating in the meeting. And thanks, Ku, again, for organising.